Right, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Dr. Anita, boleh dengar saya?
Boleh, Puan.
Okey, Dr. Chow, okey? Okey, alright. Sebelum kita serahkan majlis ini kepada Dr. Thye, marilah kita sama-sama membuka dulu sesi kita pada pagi ini dengan ummul kitab Al-Fatihah. Baik, hadirin yang dihormati sekalian, saya teruskan serahkan majlis ini kepada kita punya moderator pada pagi ini, Profesor Madya Dr. Thye, Ahli Jawatankuasa BBC UMT. Silakan Doktor.
Okay, thank you. Good morning to everyone. Kita start webinar kita yang berusaha mengurusi jawatankuasa keinstitusian Biokeselamatan dan Biosekuriti UMT, Profesor Dr. Noor Azmawati Ismail. Rasa ini dia tak hadir apa. And yang dihormati penceramah jemputan webinar Biokeselamatan dan Biosekuriti UMT 2021 siri 2 hari ini, yang bahagia Dr. Anita Anthonysamy. Dia merupakan Ketua Penolong Pengarah Jabatan Biokeselamatan dari Kementerian Alam Sekitaran Thye. Para peserta webinar hari ini yang terdiri daripada warga Pensyarah dan penyelidik UMT, pengurusan makmal UMT dan yang juga dialu-alukan peserta dari agensi luar UMT, sebab kita juga extend our invitation to a few agensi luar seperti UNISA dan Universiti Kelantan. Okay, yang turut hadir hari ini, salam sejahtera dan selamat pagi. Terima kasih diucapkan kepada para hadirin kerana webinar Biokeselamatan hari ini, siri 2 hari ini dapat direalisasikan pada hari ini. Okay, siri kedua iaitu Akta Biokeselamatan 2007 dalam Penyelidikan Bioteknologi Moden. So, tajuk untuk hari ini adalah sangat penting untuk pengetahuan semua penyelidik, terutamanya yang melibatkan biological organism. Okay. So, siri kedua ini akan disampaikan oleh Dr. Anita Anthonysamy. So, sebelum saya start seminar ini, izinkan saya give a brief introduction to all of you for Dr. Anita. Okay. Dr. Anita is currently the Chief Assistant Director with the Research and Evaluation Sections, Department of Biosafety. Okay. Her main area of responsibility is initial technical assessment and research support to process applications received for activity related to genetically modified organisms, GMO. Dr. Anita provides technical support to the Departments of Biosafety in development of policies toward biosafety and other biosafety-related issues. She is actively participates in consultants with stakeholders and biosafety experts since 2007. Dr. Anita has contributed in raising awareness about the biosafety issue through developments of public awareness materials such as guidance documents, content developments for the biosafety websites, and also represented the Department of Biosafety in giving talks about the Biosafety Act in awareness and capacity building events. Dr. Anita is the national focal point for biosafety clearance house, as well as the focal point for Malaysia in the FAO GM food platform. Dr. Anita acquired a degree in BSc Horticulture and PhD in Floriculture from University Pensyarah, Malaysia, and has a Master in Public Administration from the National University of Singapore. Okay, so Peter, I think we start the seminar. So Dr. Anita, I pass to you the platform.
Salam sejahtera, selamat pagi. Thank you very much, Dr. Chia, Puan Nur Azlina. Before I start my presentation, I just want to express my thanks and appreciation to Universiti Malaysia Terengganu for inviting us, Jabatan Biokeselamatan, to your virtual platform so that we can give a talk about our Biosafety Act. Okay, so just give me a minute here so I can share my screen with you. All right, I hope by now you can see my first slide. Okay, great. So I've titled the slide Biosafety Act 2007 and Regulatory Compliance for Researchers. So before I proceed, maybe we can go to the components of what I'm going to talk about today. So the organizers have given me 2 hours. So I will try my best to cover everything that you will probably need. So the first component that I'm going to talk about is that about the Biosafety Act and the National Biosafety Board. I will take up more time for this, and then I will zoom into items that might be of interest for researchers. So secondly, I will talk about how you can get an approval to conduct research activity. I will talk a bit about risk assessment and risk management. The fourth thing I'm going to talk about is what kind of information you need to submit to us, how to do it, and what kind of information is needed. And then the next component is really very important, is how the Institutional Biosafety Committee fits into this entire picture of regulatory component of the biotech activities. Next, I will give you some biosafety resources because you are academia. I'm sure you would like to refer, cross-refer this and that. So I can give you some references. And then finally, we will talk a bit about exemption from the Biosafety Act. So basically, this is going to be what we are going to talk about this morning. Okay? Right. So the first thing researchers need to know is what is the Biosafety Act and what is the National Biosafety Board. I will refer to the National Biosafety Board as NBB. You don't need to know everything about the Biosafety Act. At least you should know the gist of it. So the Biosafety Act 2007, okay, the objective is to set up the National Biosafety Board. We already have the National Biosafety Board, and the function of the board is to regulate living modified organisms, okay, living modified organisms and the release of products. I will explain more in my next slides. And what is the objective of having the Biosafety Act? So the objective of having the Biosafety Act is to protect human, plant and animal health, the environment and biological diversity. Okay, so this is the objective of why we are having a Biosafety Act. So for your information, the term living modified organism and genetically modified organism, in our context, we are using it interchangeably. Okay, in the Akta, it says living modified organisms. But for us, LMO and GMO means exactly the same for us in the context of the Biosafety Act. So talking about the environment and biological diversity. Okay, my slide is not moving. Okay, So Malaysia's biological diversity. So this one I minta tolong from the biodiversity section of KETSA because they have very nice and beautiful pictures of biodiversity of Malaysia. I want to let you know that Malaysia is one of the mega diverse countries. There are only 12 countries who have mega diverse are recognized as mega diverse country, and Malaysia is one of it. So you can see I have photos as well as some statistics. We have so many different types of orchids, marine fish, ferns, and even reptiles. Okay. Zooming into plant life, look at that. We have 7 of the world's 20 tallest trees. Did you know that? I actually didn't know that until I saw their slides. Okay, we have the world's largest flower. It's a bit smelly, but of course it belongs to us, Rafflesia. Okay, so look at the amount of variety that we have in the plant life. Okay, so University of Malaysia, Terengganu, I know you have a soft spot for marine life, right? Because you are very near the sea. So let's talk about marine life. Look at that. We have 6 Ramsar sites. We have 4 of the world's 7 turtle species. Okay, so the amount of variety that we have in our marine life is so fascinating. What about wildlife? Okay, we have 700 plus bird species, mammals. Okay, we have so much of wildlife that we have here. Okay, we have even reptiles, 500 plus. Okay, so these are all the wildlife, variety of wildlife. So we need to do something to protect this. So when we say protect this, protect from what? Okay, my screen is stuck again. I apologize for that. Right, so let's move on to genetically modified crop. When we mention GMO, LMO, genetically modified crop, what comes to your head? Okay, when we see something in the market that is abnormally sized, looks a bit different from how normally it would look, straight away some people might say, hey, that's a GMO, no, because it looks weird. So in your head, or maybe some people even think about this, what does a GMO looks like? We have a lot of movies, for example, like you have Jurassic Park that has a GMO monster and stuff like that. So that is actually not helping in the perception of how people think about GMO crops. Okay, maybe you think about a GM plant that probably can send you emails to water the plant. Maybe that is what some people think a GMO is. Or maybe look at this lettuce or cabbage, it has its self-protection mechanism whereby, rabbits cannot eat the GM plant. Or, a GM corn, whereby when you open the corn already it says GMO. So these are all, some fascinating ideas about GMO, but we are not quite there, okay? So when we talk about GMO and LMO, the definition that we have is a living modified organism. It's a living organism that possesses a novel combination of genetic material. Novel combination means a new combination of genetic sequence. Second point I want to make about GMO, it is obtained through the use of modern biotechnology. Okay. We also have a product of GMO or LMO. When we mention a product, it is defined by that it contains detectable recombinant DNA, whether the profile characteristics is no longer equivalent to its conventional counterpart. Okay, so these are the 2 main criteria of whether we consider something to be a product of GMO or not under the Biosafety Act. So let's talk about traditional biotechnology. Biotechnology is not something new. It is not something amazing that we just discovered yesterday. Farmers have been using it for years. Selective breeding, meaning that maybe in your farm, you have like 5 or 6 different varieties. You choose the best, and then for the next planting season, you just plant your best crop. Tak payah nak plant banyak-banyak variety. You choose your best and then you plant. So that's what we mean by selective breeding, and that is part of traditional biotechnology. What about fermentation? You know that cheese, making yogurt, making beer, making wine and all that all has fermentation involved. Your tempeh, all that. These are basic things that are used, and these are considered traditional biotechnology and is not covered under the Biosafety Act. What about crossbreeding? This can apply also for plants, but the example that I'm giving here is crossbreeding of perhaps an animal. You have one type of cow whereby the milk production is absolutely fantastic. And then you have another cow whereby the size of the cow is good. So you cross these two cows to get a progeny, meaning that a cow that has the milk production that you want as well as the size. So this is a crossbreeding in the hope of getting what you want. So this is all traditional biotechnology, which is not covered under the Biosafety Act. So if today you want to go to your kitchen and you want to bake bread, please go ahead. You don't need any approval from the National Biosafety Board. Go right ahead. This is considered traditional biotech and is not covered under the Biosafety Act. So how actually are GM crops made? So the first panel, you can see there is a problem. This GM corn is being attacked by a pest, okay? A lepidopteran pest. So there is a problem. Not all problems related to crops need GM crops, okay? Sometimes you can solve it through conventional breeding, but there are pros and cons of using what method you want to solve the problem. So let's zoom into this problem whereby this corn has an attack of pests, probably a lepidopteran pest that is attacking the corn plant. So what can you do? So you create a GM crop to solve the problem. First thing you do is identify something that can solve the problem. So in this case of this GM corn, you have your BT, bacillus, a type of bacillus that produces a protein that is poisonous to the pest that attacks the corn plant. So you have identified a protein that can kill the pest. You know what is the sequence that has this trait, meaning that what is the sequence of the gene that can produce this protein that can poison the pest that is attacking the corn plant. Okay, once you identify the sequence, you snip the sequence and take out the sequence, and then you enter it into the corn plant. So as you can see in panel number 3, this is actually a cartoon. This is a public awareness material from South Africa. So this is a very simplified version, but I will show you exactly how it's done. Okay, modern biotechnology. 2 methods that I'm showing you here, which is most commonly used. There are also other methods that you can use to insert the sequence of gene that you need into the plant. But I'm showing you the 2 most common, which is the agrobacterium method or the particle gun method. Okay, this is how you put the gene into the plant. You put it into the cells of the plant, and then you regenerate it, and that's how you get a GM plant. Okay, basically this is how it's done. So let's go back to the story about our GM corn here. Okay, so once you have gone to the lab and put in the sequence of gene that you want, then you can produce a GM plant and then you plant it. So what happens in the field when the farmer plants the GM plant produces this protein that is poisonous to the lepidopteran larvae. So the larvae will eat up the GM plant, and then it will get poisoned, and then from then it will die off. So this will reduce the attack of pests for the farmer when the farmer plants GM plant. So this is an example, a typical example of how a GM crop can solve a problem for a farmer. Okay, farmer. So other than putting a trait, perhaps a protein that is dangerous for pests, there are also other traits that you can use. You can switch off certain genes and things like that. Okay, so as I mentioned earlier, panel number 7, it talks about South Africa because I pinjam their public awareness material just to show you how you produce a GM crop. Okay, so the pathway of a GM crop from the lab to commercialization takes a few steps. This is not something that you can do in your backyard. It requires specific techniques, specific machines and equipments, and specific knowledge and specific specialization. So in the first, you need to do some R&D in the lab. How do you know which gene is the one that controls the trait that you want? All this requires some database, probably access to some database of genetic sequence, some knowledge of which gene can be used. in my first panel, you can actually see a picture of a particle bombardment gun. So that is what it looks like. Okay, it doesn't look like James Bond punya gun. This is actually a machine that you use in your lab. So once you have already obtained, probably in a tissue culture method, a plant that has the genes of your desired genes, what gene you want to put. So from then you transfer it to your plant house to see, hey, can it survive outside a petri dish? So once it survives in your glass house, then you move on to the next phase, which is probably a field trial. You take it and then you plant it a small amount in a small area. You do a field trial. Once it shows some promising result, and then you can do some commercial planting. You plant it secara besar-besaran, then you can place it into the market. Either the plant product itself, or you can have downstream products made from your product of your GM crop. So basically, this is the pathway from the lab to how it enters to the market. So there are a few steps that you can see. So let's zoom into the genetic part of what is actually happening. Conventional breeding and modern biotech breeding. Okay, so conventional breeding, you have a mixture of genetics. So the simplest example that I can give you is look at your kid. Okay, look at your children. They might have curly hair like you, but they might be tall like the grandfather. Okay, and maybe they might have brown eyes like the mother. It is a combination of traits. You cannot have a mini me. That means your child is exactly the same as you. That is not going to happen in conventional breeding. Okay, so it's usually a combination that you get at the end. It's a random gene combination. Compared to modern biotech, you already identify the gene of interest. This is the gene that I want, and then you insert the gene into the organism, and then the new variety you get has everything in addition to the gene of interest that you put in. So what is so special about modern biotech breeding and the gene of interest? So the gene of interest is not limited to what is in the gene pool. You can actually take a trait. Remember the corn example that I gave you? The gene is actually obtained from a microbe and put into a corn plant. So the taxonomic limitation of conventional breeding does not exist. You can take a gene from anything and put it into an organism. Okay, so the taxonomic barrier is not there, and therefore we want to be cautious. Remember my first slide, we want to protect our environment, biological diversity, as well as human, plant, and animal. Okay, these are our protection goal. So we want to be sure that what we have done does not have any negative impact to the environment. How can it impact the environment? You might have genetic contamination, because when you do plant biotechnology, you usually have a result that is superior compared to its conventional counterpart. Okay, so you are worried about genetic contamination. Remember my first few slides. Our variety of biological diversity is vast and awesome and great. We don't want contamination, because if our GMO is superior, there is high possibility that it can contaminate our existing biological diversity. Secondly, impact on non-target organism. Go back to my corn example, okay? Our intention is to get rid of the Lepidopteran pest that is attacking our corn plant, okay? But in the farm, okay, kat ladang, there are also other organisms that you don't want to affect negatively. Contohnya lah, you have your bees, right? Bees are present in all farm areas. You don't want anything negative to affect your bees. So when you are going to put a GM crop out there that is going to affect the pest, the Lepidopteran pest, you also need to make sure that it does not negatively affect your bees. That is a beneficial insect. Okay, so these are some of the impacts to the environment that we want to consider. It might not necessarily happen, but before you put your GM crop out there, these are the things that you want to be sure of before you put it out into the environment. We call it release to the environment. So some other examples, maybe it has potential to infect or colonize animals or plants. Maybe it can infect other invertebrates, soil microbes. Sometimes its survivability is much higher compared to conventional plant. It might behave in the same way like an invasive alien species, you know, whereby it can take over the entire population of that species. So these are some of the concerns that we have when we are talking about a GM crop and release of the GM crop to the environment. What about impact to health? So when we have a GM crop out there and perhaps later on we consume a product from GM crop, we are concerned about the allergenicity. Remember in the earlier slide I said we take a sequence of gene and we put it into the organism. But we need to check that the sequence of gene does not translate to an allergen or a toxin. It might not. We are not sure. So this is something that we have to be sure of before we approve of a product that we can consume. Okay. We are also concerned about pathogenicity. Perhaps you are working in the laboratory and then you are working with some microbes that have some additional pathogenicity traits. So these are some things that we are concerned about when you're working in the lab. Possible alteration route of infection. Perhaps this microbe, risk group 1, you know, is not so dangerous. But when you have done some genetic alteration, perhaps the conventional route of infection might change, you know. Perhaps it is by aerosol, but you know, after the genetic alteration, maybe you terkena kat tangan pun, already you get infected. So these are some things that you have to consider. Possible survival outside of the host, product contamination. So basically, this is just a list of things that we consider when we are doing an assessment, okay? Trying to go to my next slide. Okay, so let's go to the Biosafety Act. Now we are back to the Biosafety Act. Under the Biosafety Act, there are 3 biosafety regulations under the Biosafety Act. We have the Biosafety Approval and Notification Regulation. So this is a 2010 regulation. A lot of stuff that you might be interested is available in this regulation. Firstly, Regulation 5 says that you must set up an IBC if you are dealing with research and development of modern biotech. Okay, so that is in Regulation 5 under this biosafety regulation. Other than that, you have information about approval, certificate of approval, more details about notification, macam mana dah buat appeal and some other miscellaneous things. Okay, these are all in the biosafety approval and notification regulation. The other 2 regulations under the Biosafety Act is related more to enforcement. Maybe you might not be so interested in it, but if you are interested, please go ahead and read it. The first one contains offences that can be compounded and procedures to compound. The third regulation has sampling procedure. So when we go out there, when we do sampling, there's a certain procedure that we have to follow so that if you're going to take action in court, it is accepted. Okay, so these are the sampling procedures. And also for your information, we have the Biosafety Act. Under the Biosafety Act, we also have some exemption. So all this you can find in our website. So let's talk about the scope of the Biosafety Act. Just now I mentioned, I showed you the difference between traditional biotech and modern biotech. Biosafety Act covers only modern biotech. Modern biotech produces living modified organism and products of living modified organism. So you have your LMO and products of LMO. What are you doing with your LMO and product? So if you are working in a lab, we consider that contained use. Okay. So for contained use, that is covered under the Biosafety Act. If you are importing for contained use, so how does import for contained use happen? Contained use, for example, you are the researcher who produced the GM corn tadi. My story about the GM corn tadi, you produce the GM corn in your lab, so that is contained use. What if you have a collaborator in perhaps Argentina, okay? Collaborator kat Argentina tu, they are the one who produce the GM corn. Then they said, okay, we have this GM corn, you guys want to check or not whether you all want to use the GM corner ke, you want to do some physiological study ke, all that. So you said, okay, yes, let's do this collaboration with this university kat Argentina. Tapi you don't have to produce the GM con yourself. You just bawa masuk the GM con by the University of Argentina, and then you just continue the physiological studies or whatever is it that you want to do in your lab. So that is an example of a situation import for contained use. Okay, so you bring in your GM con and then you work in your lab. So that is import for contained use. You done your research, blah blah blah, and all that. Okay, nah hantar balik kat collaborator. Maybe you did some modification into the crop that you brought from your University of Argentina to say, nah, we have done some improvement. Okay, take it back and see whether you want to use it. So that is export of an LMO back to Argentina. Okay, so I'm putting it in the context of how it can work in a research situation. Once you have done some work, you want to export balik your GM crop tu kepada Argentina, that is export of LMO. So all parts of this research is regulated under the Biosafety Act. Let's talk about products of LMO. When you are releasing a product of an LMO that is regulated, import of release, because sometimes you might not do the production, but it's already produced in another country, you bring it in and then place in the market. So that is covered under the Biosafety Act. Okay. So who is regulated? All institutes and persons involved in modern biotech. Tak kira lah whether you are a public university. Sometimes private universities also have a research arm that also does some modern biotech work. So they are also covered under the Biosafety Act. Maybe you have some semi-government research institutes. They are also covered under the Biosafety Act. Tadi saya ceritakan from the lab all the way to commercialization. All stages of R&D and release is covered under the Biosafety Act. All types of organisms are covered under the Biosafety Act. So it doesn't matter whether you are working with a plant, a microbe, a mosquito, a frog. It doesn't matter what is the organism that you're working with. All types of organisms are covered under the Biosafety Act. And just now, as I explained, all types of activities. Tak kira lah you buat aktiviti apa pun, is covered under the Biosafety Act. As you can see, the Biosafety Act is so limited. Dia cover modern biotech sahaja. Only modern biotech. But if you are doing modern biotech, then the coverage is wide. Anything that you are doing, whatever activity and all sorts of organisms are covered under the Biosafety Act. It starts by being very specific to modern biotech. But if you're working with modern biotech, it is very wide. All organisms and all types of activities are covered under the Biosafety Act. Right. Okay, I can't seem to go to my next slide. Just a minute. Maybe it's because I have a lot of photos in this slide, so it's quite a big document. Apologies for this. All right, just give me a minute while I try to sort this out. Okay, let me share again. Okay, so can you see my slides? You should be able to. Biosafety oversight. Okay. So earlier we talked about the scope of the Biosafety Act. So now we talk about biosafety oversight. How is it regulated and who is the one doing the regulation? So it is regulated by the National Biosafety Board. Remember Akta Biokeselamatan we spoke about, kan? So the National Biosafety Board is the one that regulates all modern biotech activities. National Biosafety Board, which is also under the Ministry of Environment and Water. So there was a shuffle earlier. Of course, there's going to be probably another shuffle in the upcoming days, but currently it's under the Ministry of Environment and Water. That is where Department of Biosafety is. Okay, we are the operating arm for the National Biosafety Board. So biosafety oversight, who is regulating? Firstly, at your institutional level, you have your IBC. Okay, they are the ones who are regulating everything at your institutional level. On top of that, you have your GMAC subcommittee on monitoring. I will explain a bit more about GMAC, but GMAC is the technical body that is assisting the National Biosafety Board. So GMAC pun adalah dia buat pemantauan and all that for inspection. Our Department of Biosafety, we have an enforcement section. Okay, the enforcement section, they go and do inspection in laboratories. If there's a field trial, they will go and do inspection there. And then they also do monitoring in all points of entry and also in the marketplace. So Department of Biosafety does not function alone. It works together with other regulatory agencies, and then the body that is sort of bagi hala tuju and doing the management of all this collaboration with all the other enforcement agencies is the JTP LMO, Technical Committee on Enforcement. So basically, these are the four components that provide the biosafety oversight. Okay, I have moved on to the next slide. So JBK regulates everything. So as you can see, from the lab to placing in the market, JBK is a one-stop center for all matters related to LMO and modern biotech. So we regulate the part of R&D, whether you're doing field trial, commercial planting and placing of the product into the market. Everything is regulated under the Biosafety Act. Okay, so now I've moved on to my next slide, monitoring and sampling. Okay, so we do sampling and activities in all points of entry. So we go to the land borders, you know, when the products are coming in at our borders. So we do sampling there. We do sampling at seaports. We go to the ports like Westport, Northport. We go to Penang. So all these spots we go and we take samples to make sure that only approved products are being brought in, and also in airports also we take some samples from the products that are coming in. We don't work alone, especially in points of entry. We work together with Makis. Okay, so we also do sampling at the field. Perhaps we go to the farm areas, contohnya lah kat Camerons and all that. We also take sampling from the supermarkets and fresh market. So these are all areas that we take our products. So after we take the products, what do? We do some testing. Okay, we are not working alone, as I said. When masa collect sample too, we work together with Makis, or our enforcement section also goes and takes their own samples. And then we do some protein tests. Protein test is being done at the Department of Biosafety. We do have the equipments and the personnel to conduct these tests at the Department of Biosafety. So they will do a protein test. Protein test is a very simplified test. You can only test to see whether GMO ke tak. Okay, but what kind of GMO we are unable to do. So then we go to the next stage. We do the identification using the polymerase chain reaction. For some of you who are familiar, this is a test that is done in the lab whereby you can identify the specific sequence. Remember, I said in my earlier slide, how do we do a GM crop? You put in a specific sequence. So what is happening here for the identification? Identification is we are identifying the specific sequence that the GM crop has. Who does it for us? We are working together with Department of Chemistry, Kimia. We are working together with the National Public Health Laboratory. We are working together with FRIM. And also the one in Miri is not functional yet because of MCO and all that. So there was a bit of delay. But we also are going to be working together in Miri to handle the products in Borneo. So we don't have to all the time hantar ke Semenanjung. So this is what happens to the products that we have taken for sampling. I have moved on to my next slide. I'm showing you pictures of our site visit inspection. Okay. So we visit laboratories, we discuss with the researchers, and then we also see whether you are complying to any terms and conditions that is imposed by the National Biosafety Board. So these are some pictures of some inspections that you have done. These are older photos. As you can see, nobody's wearing a mask. Everybody duduk dekat-dekat, all these. So these are actually older photos that I'm showing. Due to the pandemic, we are unable to do visits as much as we would like to. For some visit, we have actually shifted to virtual visits. That is the best that we can do because we don't want to hinder research. We are trying our best. Ideally, we should go into the lab and do inspection, but due to the pandemic, we are unable to. So if we do, so this is what it would look like. We will have a discussion with the principal investigator or the researchers, and we will look at the lab, and then we will probably give you some pointers. Okay. So the penalty of non-compliance. Okay, just now we talk about the legislation, Biosafety Act. Kalau tak ada pematuhan, if you do not comply to a legislation, there is a penalty. What is the penalty here for the Biosafety Act? It is a fine of $250,000. Okay, banyak tu. So why is it so big amount? What is the big deal? Do you remember my first few slides? I showed you beautiful photos of our biological diversity. Okay. For your information, once a GM crop is out there and it happens to cause some harm in nature, in the environment, we don't have an undo button. Okay. You typing kat sana, your document, your Microsoft Word, tersilap type, tak apa, undo, right? Undo, or you just delete, settle, no problem. In the environment, there is no undo button. Dah keluar, impact is already there for your primary ecosystem, secondary ecosystem, and it goes on and goes on. Okay, so that's why we take it very seriously, and that's why the penalty is such a high amount. Okay. So I'll move on to the next component of the presentation, what you need to know. Okay, if you are going to do modern biotech activities, as I mentioned, I explained to you what modern biotech activities are just now, okay? You need to get approval from the National Biosafety Board. Okay. So there are two regulatory processes under the Biosafety Act. But for the purpose of our group here, because I think most of us are probably interested in R&D, so I will zoom into R&D matters only. Okay. So basically we have two types. One is notification, one is approval. These are the two things you have to remember. I want to do modern biotech. Do I need to submit a notification or do I have to submit an approval? How do you know? Let me tell you. It depends on the types of activity. That means at which point of research are you in? Okay, if you are in a contained use, that means kerja masih berlaku dalam lab. Or if you are import for contained use too, everything is happening for R&D in a lab, then you just submit a notification. Okay, you just submit notification, dah, settle. But just say your research has progressed to, okay, you nak tanam kat luar dah. You want to do a field trial. So then you take the leap to do submit and approval. Okay. What is the difference between these two? In your mind, you just say, okay, I am working with this GM material. I am in the lab. There is no contact to the environment because it is a contained use facility. It is enclosed, no contact to the environment, so notification. If I am planting out in the environment, there is contact to the environment, straight away, you submit an approval. It's no longer a notification. So that is how you differentiate whether you should submit a notification or whether your activity is an approval. Okay, basically that's it. So moving on, once you are very successful, lepas tu nak buat business ke, commercialize planting ke apa-apa and all that comes under approval. For your information, there are also modern biotech activities that does not involve R&D. For example, sometimes some people might sell LMO to some parties overseas, or sometimes you nak bawa masuk and then jual je. Okay, no component of R&D whatsoever. So these are the direct commercial use. These are just some examples. Maybe you are not interested, but just to let you know, these are also covered under the Biosafety Act. Okay, right. Let's go to this notification process. Tadi yang saya sebut tu. The bulk of most researchers in Malaysia currently is all working in the lab. Okay, working in the lab. So you just have to submit a notification. So applicant, which is also researchers, applicant, researcher, principal, investigator, semua sama je, okay? So what happens? What is the process of submitting a notification all the way to the Biosafety Board? Let me tell you, okay? You are the applicant, you are the researcher, you yang nak buat kerja, right? First person you should go is to your IBC, okay? You don't come direct to the Department of Biosafety, yeah? You have to go through your IBC. Submit to your IBC, IBC dah okay, IBC is the one that will liaise with us and submit your notification to us. At the Department of Biosafety, we will check, okay ke tak okay, everything complete or not. If everything complete, okay, then we will proceed to the next level. Kalau tak complete, we will contact your IBC, IBC will contact you, and then we will ask you for whatever documentation lah, maybe SOP terlupa nak hantar ke, you know, whatever lah. Once it's complete, then we move to the next phase. Okay. Once it's complete, if you are a first timer, okay, that means tak pernah submit notification. Sometimes we might want to do a pre-visit. We want to check your facility. We want to talk to you. And then you get your letter of acknowledgement. Letter of acknowledgement, bila kita dah hantar letter of acknowledgement, you can start your activity. You tak payah nak tunggu lagi. Okay, just start your activity. Why do we do this? Because we want to facilitate research. You no need to wait for such a long time. Once you get the letter of acknowledgement, we are more or less comfortable sebab IBC pun dah luluskan, dah tengok. We also already spoke to you. But if you are macam dah banyak sangat activity that is going on in some of the universities kat Malaysia ni, public universities macam-macam dia buat. We have seen their lab a few times already, so we are quite comfortable that things are progressing well in their facility. So dah keluar letter of acknowledgement, start je. Okay, you can start your activity. Meanwhile, what is happening? Your notification is going to go to the Genetic Modification Advisory Committee. I will refer to them as GMAC for short, okay? Your notification will go to GMAC. So GMAC will look at it to see okay ke tak okay. They will make their recommendation and then sampai pula ke board. Board will take a look at it. Once that is done, then they will keluarkan surat with terms and conditions. For your information, all the activities that the board has assessed mesti ada terms and conditions. Sampai sekarang lah, not even one that they say, okay, buatlah what you like. Whatever you say, we approve. Tak ada TNC, memang tak ada lah. Sampai sekarang memang saya tak pernah tengok lah anything without TNC. Everything will have terms and conditions. Okay. So they will keluarkan terms and condition and they will submit to the applicant, which is through your IBC lah. So this is what's happening. Usually the basic process is IBC, Department of Biosafety, GMAC, National Biosafety Board, keputusan dah. Okay. Tapi remember for notification, letter of acknowledgement, you can start your activity. Tak payah nak tunggu-tunggu lagi. In some cases, we might consult relevant government agencies. For example, import, you know, something like that. Kita akan minta lah. For example, you want to bring in a GM rat. Kita akan consult with DBS. Did you know that diorang nak bawa masuk rat? Lepas tu then dia kata, ada permit kat kita. Okay, like that. So that's done. Okay, so that is an example of a situation whereby we contact relevant government agency. So the entire process will take 90 days. Okay, but don't worry. Letter of acknowledgement will come to you earlier than that. In fact, last time our SOP is 14 days. Tapi masa pandemic ni, a lot of us are working from home and there are also some limitations in what we can do. So now letter of acknowledgement pun dah kurang sikit lah. If not, if everything is normal already, fully functional, usually 14 days, you should be able to get your letter of acknowledgement from the time you submit to the Department of Biosafety. you dah boleh mula kerja. Okay. This is the second regulatory process under the Biosafety Act. Okay, I've moved on to the next flow chart. I'm just telling you just in case my slides are not moving or something. All right. So back to you, the applicant, researcher, principal investigator. If you are doing research, actually approval ada macam-macam activity, but for the sake of most of us here are researchers, I will give the example approval process for a field trial. Contohnya lah, you dah masuk your transgenic glass house ke apa-apa, everything okay, you want to tanam kat luar already, okay? So you go through your IBC. Because you are going to do a field trial, there's a component of research, you still have to go through your IBC. You still cannot come directly to us. Go through your IBC. Once it's complete, then it will come to us, Department of Biosafety. Department of Biosafety also will check through your documentation, everything okay ke tak. If you're going to do a field trial, we will ask you lah, you nak tanam kat mana ni? Things like that. Okay, so everything is complete. Then we will submit to the Genetic Modification Advisory Committee. So compared to the earlier flowchart tu, kita tak nampak letter of acknowledgement, ya? Because you cannot start your activity until the entire process is done. Why? Because you are going to put it into the environment. We are not sure apakah implikasi, impact to the environment. Remember my first few slides, I was concerned that GM crops might have impact to the environment, and I gave you a list. So we need to be sure what is going to be happening when you put your GM crop outside. So until the full assessment is done, you cannot start your activity. Ini perbezaan utama. Okay, the main difference between notification and approval. You cannot start your activity until the entire process is done. So what do we do next? We pass it to GMAC. GMAC will do the technical assessment, and then they will give a recommendation to the board. Okay, so the board will look at GMAC punya recommendation. Okay, then they will make a decision. Doing a field trial, we will also consult relevant government agencies. This example that I'm going to give you is contohnya lah, if you want to plant kat Selangor, then maybe we will contact the local council and say, hey, did you guys know that they are going to do a field trial in your area? Macam tu lah. So that is the example of relevant government agency for a field trial. So now the third component of approval process is public consultation. Tadi, if you want to work in the lab, no need public consultation, okay? This component does not exist for notification. But for approval, yes, we do mandatory public consultation for the approval process. You have to go through the public consultation, and then all three components kena siap. GMAC has done their technical review. We have contacted the relevant government agencies and gotten their feedback. Public consultation dah selesai. All three done, barulah board will review. After the board has reviewed, then they will give their decision. After you dah dapat sijil, okay, we have a certificate of approval, then only you can start your field trial. So for researchers, I want to tell you, this is the process. If you intend to do a field trial and then apply earlier, okay, you don't want to kacau your timeline. So if you don't want to kacau your timeline, work backwards. Work backwards on how long you need to get an approval from the National Biosafety Board. Supposedly, it's supposed to take 180 days. Tapi buat masa ni adalah beberapa kekangan, okay, in terms of turnover rate of board members, and then also, shuffling of ministries and things like that also causes a bit of delay in the board meeting to do the review. Okay, so but more or less it's supposed to take 180 days, and because of the pandemic as well, there has been some delay in all our processes under the Biosafety Act. Okay, let's zoom into the public consultation, which I believe that you might be interested to know how public consultation is done for the Biosafety Act. So you need to do an announcement. On the right side of the screen, you can see this is an example of how an announcement looks like, okay, for placing in the market of a GM corn for the purposes of food, feed, and processing. Okay, we will put an advertisement in 4 main newspapers, 4 languages, Malay, English, Mandarin, and Tamil. Okay, 4 languages, and it has to be in a national circulation newspaper. You cannot put Bulletin Terengganu macam tu. So orang Terengganu je boleh baca. Okay, orang kat KL, Penang semua tak dapat. Okay, cannot. So the newspaper of your choice must be a national level circulation newspaper. Okay, that's one. Number 2, you have to do announcement twice, meaning that today you do announcement, 2 weeks later you do an announcement. Okay, so each newspaper 2 kali, total 8 announcements have been done in the newspaper. Okay, this has been going on for a while. It is quite conventional. I know everybody is moving online. Is this effective or not? It is quite questionable whether people are reading announcements in the newspaper. But slowly we will evolve in terms of how we do the public consultation. Other than doing the announcement, we also have our stakeholders. You know, we have a bunch of stakeholders, environmental NGOs, about 10 to 15 of them. We will personally send an email to them to say, hey, this public consultation is taking place. Do you want to give your comment? We also send email to all IBCs. Whenever there's a public consultation going on, we will send also alert to IBCs, see whether anybody want to comment or not. Okay. So public consultation, the cost of doing the announcement in the newspaper is borne by the applicant. I want you to take note of this as well. So earlier I said, plan your timing. Kalau dah tahu nak buat field trial, submit your application earlier. Okay, that is in terms of timeline. Now I want to talk about your finances. Okay, usually research, you punya dana, your research to conduct is usually limited. So when you are doing a proposal for a modern biotech activity, please take into consideration how much you are going to spend for the public consultation to do announcement in these 4 newspapers twice. Okay, put that into your costing. Okay, regulatory compliance for your research activity. With that, then you tak adalah kacau your, you know, your allocation, nak beli your thermocycle ke apa-apa kan, or nanti potong gaji for your research ke, tak adalah macam tu, right? So you already put the cost into your budget. What is the cost of regulatory compliance? Okay. Another thing you have to do, which is different from your notification, is you have to prepare a fact sheet. We will tell you how to prepare a fact sheet, and then with that, we will do a public consultation. Okay. You cannot just look at the announcement. It just says, okay, this is going to go in the food, feed, and processing. That's it. You have to remember the cost is borne by the applicant. If you want to give too much details in that small area, it's going to be more. Okay, it's going to cost you more. So what we do is just a basic announcement. Public consultation is taking place. The fact sheet contains more information about what is going to take place and also about the GM material that is going to be released. So basically, this is a flow chart. Okay, you, the applicant, will inform us, okay, I want to do my public announcement hari ni, and then 2 weeks later, you give us the date, kita tengok kesesuaian. Because for your information, we do not do more than 2 public announcement per month. Okay, if not, then it will be too many things happening, and then we give proper attention to the announcement. Maximum 2 kali sebulan sahaja. So ada queue lah. So then we will have to discuss which date is appropriate for your public announcement. Okay. And then the first day that your public announcement masuk surat khabar, JBK will upload your fact sheet in our website. Okay. We will upload your fact sheet on the website. So anybody who see the announcement will go to the JBK website, see the fact sheet, find out, oh, this is going to take place. Oh, this is the site, blah, 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 whatever lah. They find out about that. And then once it is done, you have to provide proof of announcement. That means you have to provide us a scanned copy of the announcement. Public consultation will take place for 30 days. For 30 days, we accept all input from public ke, NGO ke, whoever lah nak bagi komen, we will accept for 30 days. After 30 days, we will tutup. unless more than 30 days is needed. If it's anything that is contentious, it probably needs more than 30 days or something like that, then we can extend it for more than 30 days. I'll give you an example. We did a public announcement. Sometime in March last year. Okay, that is when the pandemic first started. That we closed down a lot of things. We also closed down our government agencies, and suddenly everything was not normal anymore. So the public announcement that was taking place, we extended it for 45 days because we take into consideration that a lot of changes have taken place. People do not have time to take note of it. NGOs also, don't have time and all that. So that is an example of how we extended from 30 days to 45 days for those two cases. It's not because it was contentious, but because of the situation that we were in. Okay, right. So once that is done, after 30 days and blah, 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 we will consolidate and then we will report back to the NBB. So if you notice study tu kan, there are three components of approval. First is GMAC assessment, of which I will talk about in my next few slides. And then you have your component of public consultation, and then also your regulatory agencies. Okay, so these three components are very important for the board to take note. Okay, so maybe you are not interested to do a field trial lah, contohnya lah. But you can also give comment, because I believe most of us who are attending today are researchers. You have the basic knowledge about genetics, and you also have the intelligence to know what is happening when you are doing a public consultation that deals with a GMO. So I would invite you, please participate in a consultation. Currently, there's no consultation, but you can join our Facebook lah, Twitter lah, Instagram lah, apa-apa lah. We have our social media, and my last slide, I put everything for you. Just join, and then kalau lah ada public consultation opportunity, you'll be alerted through all our social media platform, okay? If you go to our website, you can see there's one tab specifically for konsultasi awam, okay? So if you go to this website, if it is taking place, go to konsultasi semasa, you can see like this, okay? You will see at the bottom of my screen, konsultasi semasa. What is the event? That means what is the GM that is going to be released? Who is the applicant? Just say you are the researcher going to do a field trial, then it will have your name lah. Universiti Malaysia Terengganu, nombor rujukan, JBK, blah, 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 macam tu. Then after that, we will also put, tadi remember you did the announcement in your newspaper, Keratan Akhbar, we will also put here. And then this is the fact sheet that you prepared. Okay, the fact sheet that you prepared will have a lot of information. And then the status. Usually we have a status whether it's ongoing, and then it will give you what is the final date that you can submit your input. So all this you can find on our website. Okay, go to the fact sheet. Before you comment, when I tell you jagung DP202216, what on earth do you know about this jagung? You don't know, right? So you have to go to the fact sheet. So this is the fact sheet. So when you open the fact sheet, what do you see in the fact sheet? Okay, this is a contoh of the lembaran fakta. By the way, our website is 2 languages, English and Malay language. You will be requested to do a fact sheet in also 2 languages, Malay and English. Okay, this is to cater for people who might not be very well versed with either language. Then we give the opportunity for them to have the information in a language that they can understand better. So 2 languages. So check, the fact sheet will have purpose, description of the activity, assessment of the possible risk, emergency response plan, semua ni ada in your lembaran fakta. If you're interested, you can go to our website. We have an archive. All the previous fact sheets are all there. So look at the fact sheet first and understand what is happening. Straight away, don't say, tak nak, tak boleh, or, those are not very useful for the board. Check and see what is happening, what is the event. And then if you want to comment, go ahead and comment. And send your input to dob@biosafety.gov.my. Okay? You cannot give your nama kesayangan, when you want to submit input to the Department of Biosafety. This is a formal process. We cannot masa mesyuarat board meeting, then we say, X-Men 457 said that, you know, this, they are concerned about the allergen in the GM crop. We can't do that, okay? We need your full name and your contact details, okay? Please give your proper name and your contact details. And your comments or your queries can be about the GM crop, can be about the area that the field trial is going to be conducted. You know, it could be anything. So you have to contribute to the board's assessment. You cannot just give a one-liner, I tak suka, like that. How is that going to be useful for the board's assessment? It is not. Okay? It's not limited. For public consultation, it is not limited to be science-based. Okay? It can also be other things such as cultural and all that, because under Section 35 of the Biosafety Act, if needed, we can also take into consideration socio-economic consideration for the board to make a decision. It is possible. So you don't only have to comment for the science-based stuff. You can also have other things that you are concerned about. You can also give your input to the board. Okay, that's our email address. Okay, let's go back to the regulatory process just now. Got two different regulatory processes for approval. These are the fees. Okay, when you're going to submit your approval form, these are the fees. You are going to do a field trial. The fees depends on the size of your field trial. Okay, but if you are going to submit a notification, there is zero fees for notification. So tadi, if you are going to do a field trial too, so these are the things that you have to consider lah. One is your public announcement punya costing. Another thing is masa nak pemprosesan form, you also have a fees to consider. Okay, so these are the things you have to remember when you are doing your budget for your research activity. Okay. For notification, don't worry, we won't cut into your budget because currently for every notification that you give, we are not charging any fees. We will just process it. So please submit lah beramai-ramai notification because tak ada fees. Okay, no cost incurred to you. Okay, so decision making under the Biosafety Act. I have moved on to my next slide. Decision making under the Biosafety Act. How do they decide what to do with your application or notification that comes in? First, they will look at the technical advice provided by GMAC. So GMAC will just give technical advice lah, ini blah 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 whatever lah. So everything is technical. They will also look at the policy advice provided by relevant government agencies. Okay, I gave you some examples earlier. Thirdly, they will consider public consultation input. As I also explained in my earlier slide, maybe this is NGO ke, people living in that area ke apa-apa lah. And then public consultation, remember it is limited only for release activities. If you are working in the lab, this does not apply to you. Okay? Next, socio-economic consideration. Kalau perlu, it's on a case-by-case basis. If we need to consider, we will consider. If it doesn't have to be considered, then it will not be part of the assessment. Dah buat semua tu, then the board will make a final decision. So as I mentioned just now, the pandemic has cut into our usual processes, and then we are not really stopping work in terms of the regulatory process under the Biosafety Board. It's just that we have learned to evolve and adapt. So right now for our GMAC, we have done like online meetings. We go through the applications online. We are doing our best. Because it's being done online, you know, sometimes it takes a bit longer because of technical glitches and, you know, all these kind of things. But work is still progressing for your information. Okay, so what are the decisions that the board has done so far? Okay, these are the things that have been approved for release so far by the National Biosafety Board. Food, feed and processing. There is a large component of our approval of the board for release. Mostly we have different types of grain. We have also finished products that are placed in the market. Does Malaysia have field trials? Yes, you will be surprised. Okay, so in the year 2010, IMR conducted a mosquito field trial. So memang kecoh sekampung lah, this GM field trial. So actually this was the very first case under the Biosafety Act. And it was the first thing that we processed, and all eyes were on us. Okay, so that field trial was finished. And then we also have a field trial on papaya as well as rubber. So these are the field trials that the board has approved. And then for commercial planting, we do not have commercial planting. Like some countries, they have a moratorium. That means dia tak benarkan sama sekali for commercial planting. So for Malaysia, we don't have commercial planting, not because there's a moratorium, but because currently there are no requests. So being in the Department of Biosafety, we can see the research that is being done locally by our researchers. Currently, there are some at the stage of field trial, but not many. Some are still, still tweaking the research in the laboratory stage. So currently, there are no requests for commercial planting. Okay, let's look at some statistics, okay? So for approval, we have 59. And then for notification, we have processed about 240 plus notifications. So the one in yellow, you can see notifications. The one in sort of like blue, purple color too, that one is our processing for approval. So as you can see, the bulk of what we are working on is mostly contained use activities. Okay, I have given you stats for 2020 sahaja. So we have about 17 notifications that we were processing since 2020. Okay, so these are the stats for Malaysia. So for your information, a lot of things you can find on our website. Tadi saya katakan, if you're doing a release, you can find information about the GM, siapa applicant and all that. For notification, go to our website. I challenge you. You cannot find anything about notification. Okay. Currently, we do not put any information about a notification that we are currently processing or even previous decision of the board on any content use activity, any research activity that being done in the lab, memang kita tak keluarkan for public access. Okay. We have done a consultation with researchers. They said, ayah, tak maulah, don't tell anybody what we are doing. Then we ask, tajuk tak apa ke? We just letak tajuk je. Dia kata tajuk pun tak boleh, nanti orang ikut. Nah, macam tu, tak boleh lah because they haven't published yet or what lah. So for that reason, in respect to researchers who were not happy if any information about notification is put out into public access, currently all information is with Department of Biosafety, is with your respective IBCs. But other than that, we don't put it out there. If you yourself do publication, it's up to you lah kan. If you yourself put it on your UMT punya website, okay, these are all the research that is being done under UMT. Banyak betul kita punya research modern biotech. Sukati you lah, okay. But you cannot get it under the Department of Biosafety because we are respecting the wishes of researchers. There will be no information of notification. So when you are submitting your information, rest assured that it is not available for public access. Right, let's go to the types of crops approved for the release. Okay, from 2010 to 2020. I have moved different slide now. Now you can see a pie chart. So as you can see, most of the crop that are coming in for approval from the National Biosafety Board mostly is soy and corn. Other than that, we also have cotton, potato, oilseed rape, papaya lah, rubber, semua ada. Okay, but the bulk of it is corn and soy. Okay, so I want to give you a summary of the regulatory process. Okay, under the Biosafety Act, there are two regulatory processes. First, notification. Secondly, is approval. Okay, you just remember this jadual, okay je. Okay, purpose of notification is continuous. That means semua kerja dalam lab, you submit a notification. If there's any contact to the environment, if you're doing a field release, placing in the market dan sebagainya, you need to submit an approval. So that is considered a release. Okay, timeline for notification under the legislation. Nak process notification is 90 days, approval is 180 days. Tapi notification 90 days ni not so much of an issue. Okay. Consult public. If you're working in the lab, do you need to consult public? Tak perlu. No need to consult public. No public consultation component for the processing. If you're going to release into the environment, yes, mandatory public consultation. That is one component that the board will assess before it makes its decision. Let's talk about fees. Notification, you want to work in the lab, zero. Okay, no fees. Submit lah beramai-ramai. Okay, no fees. Okay, but for approval, as I mentioned earlier, if you're involved in research, it depends on the size of your field trial. For any commercial kind of application, it is 5,000 ringgit per application. So when can you start your activity? Selepas submit notification, you get the letter of acknowledgement from JBK, you can already start the work in your lab. Okay, you can start the work in your lab. But if you're going to do a field trial, anything that involves release, do not start your activity sehinggalah until you get your certificate of approval in your hand. Then only you can start your activity. Okay. So as in any process in Malaysia, okay, under any other legislation also, kalaulah kita tak puas hati lah dengan dia punya board punya decision, ngapalah dia decide macam ni, tak apa, jangan bimbang. Okay. You can still appeal to the board. You can appeal to the minister, sorry. There is a process to appeal to the minister if you are not happy with the decision made by the board. So for the notification process or the approval process, there is a provision under the Biosafety Act to do any appeal. Okay, so maybe I will stop sharing my screen for a while and go back to the organizers whether you want to take a short break because we agreed to do a short break after 1 hour.
Yeah, thank you, Dr. Anita. All participants, we have 5 minutes short break. During this time, if you have any question, you can ask Dr. Anita. Okay, so but before that, I think maybe I would like to highlight a few points for all of you. So Dr. Anita actually already explained to us quite comprehensively on what are GMOs. Okay, so basically refer to modern genetics, modern biotech. So maybe all of us researchers, we can analyze or go back to see whether our research fall under this modern biotechnology, which cover under biosafety regulations. Okay, so and Dr. Anita also highlighted the enforcement. So under this enforcement, because I think I would like to highlight to all of you also, because there's a penalty. I think many researchers, we don't know there's a penalty for non-compliance. Okay, so if we the penalty is very high, 250K fines and imprisonment of 5 years maximum. So I think we all researchers that involved in this modern biotechnology, we need to aware of these regulations. Okay, so other things like maybe the approval notifications, and if you are not sure about your research, I think you can always talk to IBC UNT. So you can talk to either Professor Noor Aznawati or Azrina. So Azrina is our very important person here because he organized everything. He conduct because all approval, because I have gone through the notification step for my own transgenic research. So actually it's quite tedious. Many things you have to fill out until at the end you have, when you dispose, how you dispose all of your transgenic organisms in the lab. All you need to have the records. So I think all researchers have to aware of this process. So if you are not sure, talk to IBC UNT. Okay. So do you have any questions? You can just on your mic and ask Dr. Anita before she continue her presentation. Any questions?
Dr. Anita, I have one question posted to me. You mentioned about the field trial and the continuous. Okay, for the continuous is very clear. It is conducted in the lab. What about the experiment that is conducted in the glass house, okay? Which regulation does it fall under, and what are the requirement of or specification of a glass house that handle the experiment of the GMOs? Okay, Doctor.
All right, thank you for the question. So if your material is already at the stage of going to the glass house, so how is your glass house? Is your glass house fully contained? If it is fully contained, meaning that there's no contact to the environment, it is still an enclosed area, then we would consider that notification. Okay, and perhaps if you are, just say you are planting in a glass house, okay, but you are planting in the soil in the glass house. Contohnya lah, because some glass houses is not cemented surface. So just say you have soil, it is a glass house, it is covered outside, but your base is soil, so that will fall under approval. So what is your cutoff point? Whether I should submit a notification or whether I should submit an approval. Does it involve any contact to the environment? Okay, if there's no contact to the environment, then it would be a notification. However, if there's any contact to the environment, just now I gave you a unique example whereby they did planting in a glass house, but they were planting it in the soil. So part of the assessment is we also want to consider, will it impact our soil microbes? Because we do have some nitrogen fixing microbes and all that, we just want to make sure that everybody is fine under the soil, right? So that is another thing that we want to consider. But basically, most glass houses, their base is cemented and there's no contact to the environment. The integrity of your glass house is very important. So you were asking apakah ciri-ciri, right? Like the criteria of the glass house, meaning that if we assume that it is a contained use facility, then we need to ensure that everything is done to keep it so. For example, if you're working in the lab, situation is different. When you are going outside, it's also different. People walk in and out, especially glass house. Accessibility to glass house lain sikit, because you might have your general workers to come and do some cleaning up, or maybe you want to wash the surface of the wall ke apalah, whatever lah, so not necessarily researchers alone are assessing. So you need proper procedures, accessibility to the glass house. Siapa yang boleh masuk, siapa yang boleh keluar. What is being done in the glass house? Most glass houses, they have like an area. Before you masuk, ada one area, like a double door. Okay. So you masuk je, then maybe you change into another outfit ke, tukar kasut ke, whatever, then only you masuk. So that is actually one of the feature that I would highly recommend if you do intend to have a glass house at UMT, So it will be a protection so that whatever's inside is not going out. Accessibility is important. Who has access to it is important. How you manage things in the glass house is important. For example, if it's already at a stage where you are planting kat glass house, obviously the plant is also bigger. Then maybe you want to trim ke, crop ke, those kind of things. But you have to remember, yes, you already crop the leaves of the plant, but that is still a GM material. How do you dispose of it? Even though it's in the glass house, okay? So how do you dispose of it? So then we will look at your SOP. Okay, you're planting in the glass house. How are you going to dispose of it? So what is the normal procedure to dispose of it? If it is, because I have worked in a glass house before, so I will just give you a plant example. So once we do the trimming of the plant, we will just buat macam sampah biasa. Okay, we will put in the garbage bag and then we'll put it outside. Then the lorry will come and take all the sampah, bag sampah of all the planting material, and then they'll throw out. That is a normal procedure if it's a non-GM plant, okay, the basic procedure. But if it's a GM plant, maybe you need to incorporate an SOP whereby you bring it back to the lab to autoclave, to make it non-viable, so that when it's gone outside, it doesn't replicate. Then you thought you buang already, then tengok, there's a plant that is growing outside there that's actually a GMO, right? So those are the things that you should avoid. So that would be, in my opinion, a little bit of differences between how you handle an activity in a lab and how you handle activity in a glass house. I hope I addressed your concerns.
Clear, Doktor. So kena ada specific requirement dan juga SOP how to treat the GMO plants conducted in the glass house. Dan dia sepatutnya separated lah kan daripada non-GMOs, very contained specific untuk dia sahaja. Thank you, Doktor.
Thank you, Azina. Any other question from participants? If no, maybe I may ask one more. Dr. Anita, just now you during your presentation, I think in Malaysia there are many GM products have been approved, right? So how about the labeling issue, labeling of the GMs? Is there any regulations on labeling in the
Yes,
market?
yes. Thank you for the question, Dr. Cha. I have not included labeling in my slide deck because I thought that, you know, for this topic today, I will just focus on researchers. But thank you for the topic. We do have a labeling requirement. So for products of GM, countries can take different approach. Some of them say no need labeling. Some of them say voluntary labeling. And some go for the approach of mandatory labeling. Under the Biosafety Act, we have gone for mandatory labeling. That means if you are putting a GM product in the market, it must be labeled as GM. So under the Biosafety Act, it says labeling is mandatory. But what are the details of how you label? Okay, so the details, for example, if it's a food product, we have a GM food regulation under the Ministry of Health. Okay, you have a Food Act under the Ministry of Health. Under the Food Act, there is a specific regulation on how to label GM products that are food. Okay, so that one I didn't go into detail, but there are, I can give you some links later on if you are interested to know how it's being labeled. How is it being enforced? That is being done by the enforcement officers under the Ministry of Health. So they will be the one checking the product, whether it's labeled or not, is it a GM or not. Okay, so the Biosafety Act, we will tell you whether the event is approved or not. Let's go back to the example of GM corn. So we said, okay, this GM corn, DP22016, is approved for food, feed, and processing. So the board has made a decision, then that's it. So from the corn, just say you decide to make cornflakes or your corn biscuit, okay? So who is regulating the product of cornflakes or corn biscuits? That would be the enforcement under the department, under the Ministry of Health, because they are the ones who are labeling, they are the ones who are regulating all sorts of labeling for food products. So remember, I said the Department of Biosafety does not work alone. So this is an example of a collaborative activity that we have with the enforcement under the Department of, under the Ministry of Health, more specifically FSQ, Food Safety and Quality Division, under the Ministry of Health. Was that okay? I think Dr. Cha?
You are back. Okay, thank you for your explanations. But because until now, actually, we are hardly know. I think we still never see the labeling of any GM products in the market. So maybe they are not. I don't know. So they are not labeling or they are labeling. So that's
So
why we don't know.
if you look into the details of what should be labeled and how it should be labeled is found in the regulations, as well as there's a guideline. So for your information, there is a threshold. Threshold is in the product. For example, if I make a corn biscuit, right? So maybe my ingredients comprise of sugar, flour, and all that. So based on these ingredients, there's a threshold of how much of the GM product is inside there. If it's above a certain percent, then it gets labeled. If it's below a certain percent, then it does not need to be labeled. So if you look at the guidelines, then you can understand better how they decide whether or not something should be labeled or not.
Okay, thank you for your explanation. So if there are no other questions, so we invite Dr. Anita to continue your presentation.
All right, thank you. I will continue my presentation. Okay, so Azlina, you can see the slide. It should say number 3, researchers need to conduct risk assessment.
Yes, you can see the slide.
Okay, all right, thank you so much. All right, so the third thing I want to emphasize is that we as researchers, we need to know how to conduct a risk assessment of whatever activity that you are doing. And from the assessment, you need to develop a risk management mechanism. Okay, so these are specific provisions under the Biosafety Act. If you are interested, you can look at Section 36 of the Biosafety Act. It specifically talks about risk assessment, risk management, and finally, emergency response plan. Okay. Right, it's stuck again. I'm trying to move my slides. Please be patient with me. Sorry, I really don't know what's happening. Try to share again. Just a moment here. Let's move on to the next slide before I share. All right, now you should be able to see a flow chart on your screen. It's not. Can you see a flow chart on your screen?
Yes.
Okay, great. Right. So this is an overview of a risk assessment process. Okay, when I spoke about risk assessment, we have a 3-tier risk assessment. That means 3 stages of risk assessment is being done. You as the applicant, principal researcher, okay, you are the one who are doing the activity. You are the one who knows what kind of risk is involved in the activity that you are doing. So with that, you will provide the initial risk assessment. Okay, you are the one who proposed the risk assessment, risk managed plan, and emergency response plan. And this will be filled up in the form that Dr. Cha spoke about earlier. After you fill up the form, you will submit to the IBC for review and endorse. So kalau IBC tengok say, okay, I think your risk assessment okay, your management okay, everything okay, then that is the first layer of assessment. At your institutional level, they have done an assessment. Okay. And then they will submit to the JBK. So JBK will look at it and say, okay, you have your risk assessment. Okay, in your risk assessment, you said you dispose accordingly, okay? You guna your glass house and then you punya pokok you kata autoclave. Where is the SOP na autoclave? Saya tak nampak. So JBK will say, please send us your SOP for autoclave. How you're going to autoclave the material. So that is your request for additional information. And then you will give us the SOP. Okay, so we are happy already. Then we submit all the documents that you gave us to GMAC. So GMAC is doing an independent risk assessment. Why do I mention independent? Independent because IBC dah buat review. Tak semestinya GMAC does the same kind of review. Okay, it's different because IBC is a different group of people. GMAC is a different group of people. So maybe IBC did not think that this component is important, but GMAC thought, no, this is very important. They need to inform us about this. Okay, fine. So it is an independent risk assessment. So just now you were working in the transgenic glass house. Then maybe I give you an example with an animal. So IBC review, they said, okay, these are the team members, everybody okay. Tapi masa GMAC kata, where is the training record of all the researchers all the team members who are involved? We want to know because this one is, in our opinion, a high risk punya activity. We want to see what is the training that was done by the researcher who is going to be handling it. So then request of additional information. We ask, can you please provide training record? And then it will go back in the loop. Come back to JBK, JBK give to GMAC, GMAC look at it. Yeah, okay, everything is fine. Okay, then we prepare our assessment and give to the board. Okay, then the board will look at it and then they will make a decision. Board can also request additional information. This is quite rare because by the time IBC is finished with you, by the time GMAC is finished with you, by the time it goes to the board, dah tak ada benda lagi nak tanya. Everything dah ada dah. This one, okay, everything got complete already, usually lah. So very seldom the board will request for any additional information, but they can ask if they want to. Okay, it is a provision under the Biosafety Act. Dia nak tanya pun boleh je, but very rare lah. Memang jarang sekali lah the board will ask any questions because at that stage already mostly it is as complete as it can get. Okay, so if you look at my red color arrows, it can come from JBK, it can come from GMAC, it can come from the board. Request of additional information can come from 3 different people. In fact, your IBC also can ask you additional information. They are entitled to finish their review. So just say JBK tadi tu ask you for, okay, what is your SOP for autoclave? You tak bagi, okay, then you bagi already. Then GMAC tanya, what is the training record? Okay, dah tanya. But we are asking you 2 different things. You as the applicant say, kan that day you asked already. Why you ask me again? And again, and again. Okay, it's not asking again and again. You must understand this different group of people who are doing the assessment and checking whether your information is complete. Okay, so we are making the best of our efforts to make it as complete as possible so that when it comes to the stage of the board assessment, there's nothing else left that they can ask. Okay, so actually board meeting is like Viva lah, because you all are university, right? So you all will know how is the situation like. Okay, so masa board tu, board tanya soalan ni, then our pengerusi GMAC will say ada. This one say what did they consider this? Ada macam Viva lah. So department also will answer on your behalf. GMAC pengerusi also will answer on your behalf because you are not there at the board meeting. So we want to be as prepared as possible. So then before sampai board, we will ask you lah everything to make sure it's complete as possible. Don't be offended, don't be frustrated, don't be exasperated. It is all for the best, to get the best possible result that we can get from the board punya decision. Okay, once the board has decided, then you have your terms and conditions as I mentioned, and then monitoring by the Department of Biosafety and monitoring also by the IBC. Remember my earlier slide, I showed you about biosafety oversight. Either it can be our enforcement, it can be the inspection group under GMAC, it can be by your IBC. So all these people can come and check on you and monitor your activities. Okay, right. So who is this GMAC that is looking at your form? What are they looking at? So GMAC is a technical committee. They come from various backgrounds because for the notification, as you remember the scope of the Biosafety Act, mosquito lah, plant lah, microbe lah, macam-macam kita cover, right? So GMAC also needs various expertise. So we have expertise in virology, plant breeding, entomology, animal breeding, macam-macam lah. Each one has their own expertise. We come together and we do an assessment and then we make a recommendation or technical advice to the board. So basically they will just do technical assessment. Okay. I've moved on to the next slide. So now you can see the National Biosafety Board. Okay. Who is the chair of the National Biosafety Board? National Biosafety Board pengerusi is Sekjen. Okay, KSU of Ministry of Environment and Water. He is, or she lah, but right now it is a he. He is the pengerusi of the National Biosafety Board. Okay, Secretary of the National Biosafety Board is my DG of the Department of Biosafety. So keahlian, okay, membership of the National Biosafety Board are people who are main stakeholders for modern biotech in Malaysia. Who are they? Ministry of Health, Ministry of Plantation, Ministry of Domestic Trade, Ministry of Agriculture, Ministry of International Trade, Ministry of Science. Okay, so you have these 6 ministries who are members of the board. On top of that, we have also appointed 4 experts. These 4 experts, maybe they have a science background, maybe they have the technical expertise, and maybe they have the experience. Okay, there are certain criteria for the experts, but they might not necessarily be policy people. So the 6 ministries, sometimes they have science background, sometimes keahlian is by dia punya PUU lah, dia punya lawyer yang jadi ahli. So depends, whoever can take into consideration the policy matters of the respective ministries. But the experts, it could be someone from a science background. So that's why I said earlier, maybe the board also can ask you for additional information. So this is the setup of the National Biosafety Board, and they are the ones who are looking at your notification or approval form. Okay. Now I've moved on to my next slide. I am going to show you a risk assessment matrix. So Dr. Cha has the experience of filling up Form E. When you fill up Form E, there will be one component of risk assessment. So we will give you lah this table you kena isi. So when you are working in a contained use facility, these are the components you should consider. Signs of the genetic modification, admin policy, containment integrity, and finally the component of special risk unique to notification. Basically, you have to fill up this table and submit to us. How to do a risk assessment is actually a skill, and also it is also a different training that the Department of Biosafety provides. Unfortunately, we do not have the time to do that. So I just want to show you this is how a table would look like, and you have to go through the process of risk assessment. Okay, so these are the components that you have to look at when you are doing a continuous activity. Once you have identified the risk, you propose how you are going to manage the risk. Okay. For example, signs of genetic modification. I will say that, okay, I'm working with this microbe and it's quite a dangerous microbe, and I need to manage it. So what is my management strategy? So my management strategy, I will say that all work will be carried out in a biosafety cabinet so that aerosol does not reach the one who is handling the experiment. Okay, so this is just an example, all right? Okay, so now I have changed my slide. Let's go to the biosafety containment. So under WHO classification, you have, if you're working with a microbe, you have your risk group 1, risk group 2, risk group 3, and risk group 4, right? So that would be your conventional classification of a microbe. So sometimes you might think, okay, because it's risk group 2, it should go into BSL2. That is not necessarily the case. Or if it's a risk group 1, it should go in a biosafety level 1. So for some of you who are not familiar, laboratories have various classifications. You have your level 1 up to level 4 with different specifications of the laboratory to handle material that have different types of risk. So the lower your BSL, okay, the lower the risk of the material that you are handling. You go from BSL 1 to BSL 4. In Malaysia, we do not have a BSL 4 facility. The highest that we have in Malaysia is only BSL 3. Right. So now you are doing the risk assessment. Okay. So the risk assessment is the agent characteristics, the project objectives, what is the sequence that you put in. Because remember in my earlier slide, when we were talking about genetic modification, I told you that the sequence of the gene is not limited to taxonomic barriers. So for example, if it's a very harmless microbe that belongs to a risk group 1, I can easily put a sequence that causes the pathogenicity from a risk group 3 pathogen masuk in my E. coli K12. Then my E. coli K12 is no longer as harmless as I thought it was, right? So that is the difference of a genetic modification experiment compared to a usual experiment that you can do. So these are the things that you should consider when you are doing the science of genetic modification risk assessment. Okay, so risk assessment of the activity is done. So where do you want to work? It should match the risk assessment. Say, oh, this is very low risk lah. You can actually do it in a BSL 1. No problem. So the risk assessment must match the premises. For example, I take an E. coli K12, which is quite harmless. We know quite a lot about E. coli K12. Huh? So then suddenly I put one very dangerous punya sequence into E. coli K12. Maybe I cannot recommend that this E. coli K12 transgenic can be done in a BSL 1. Maybe I will recommend through the risk assessment that I did just now. This activity must be done in a BSL 2 or BSL 3. It cannot be in a BSL 1 because of the insertion that I put in, right? The sequence that I put in makes this E. coli K12, this transgenic version of it, not as harmless as the conventional one. So I cannot allow it to work in a BSL 1 because it can harm the person who's handling, it can harm the other people who are using the lab, so on and so forth. Okay, so the main point that I want to make here is, first you do the risk assessment, and where you are going to conduct your premises must match the risk assessment that you have done for your activity. I have changed slide. Now we will talk about risk assessment, short form RA, risk management RM, and your emergency response plan. Okay. So you might, IBC might say, okay, this is the risk management measure dah cukup. Tapi bila keluar board punya decision, maybe they have additional risk management that you need to take into consideration. So whatever is it, finally you ikut siapa? Ikut hati, ikut IBC? No, you have to follow whatever the final decision of the board makes. Okay. Non-compliance, there is a penalty. Yeah? So whatever terms and conditions that the board imposes, it is compulsory, mandatory for you to ikut. You tak boleh suka-suka, you tak suka lah, tak nak lah. Cannot, okay? There is a penalty. You tak ikut, okay, you have to face the charges of non-compliance. Okay? Not only for your risk management, but when we look at your emergency response plan, you should also do exactly what you propose to do. Your emergency response plan punya lah cantik. Okay, when something happens, this is what we are going to do. Tengok-tengok masa emergency tu, what to do ah, what to do ah. Okay, everybody panic gila like that. It doesn't work that way, okay? So, before it happens, you must already have a plan on what you want to do. You cannot bila benda tu berlaku, okay, emergency meeting, what shall we do, what shall we do, okay. If you are going to handle GM material, unfortunately for you, this is not possible, okay. If you are going to handle modern biotech activity or GM material, your emergency response plan must already be in place, okay. Okay, so Azlina, I'm going to play the video now. If you think that it is not working, then you just stop me, okay? So I want to show you a video. This is from Monsters Inc. So to let you know how well prepared, although this is a cartoon, you can see everything is already well prepared. You won't see this video, dia cepat-cepat tengok dia punya SOP ke apa-apa, tak ada. Everybody knows exactly what to do.
Hey, Wazowski, nice job. Those numbers are pretty sweet.
Are they? You know, I hadn't even noticed. And how is Georgie doing?
He's doing great. I love working with that big guy.
Keep the doors coming, Charlie. I'm on a roll today.
George and I are like brothers. 2319! We have a 2319!
George Sanderson, please remain motionless. Prepare for decontamination.
Oh, not the CDA.
Watch yourself. Stand back. Careful. 990, ready for decon. Hey, thanks, guys. That was a close one. Okay.
Okay, people, take a break. We got to shut down for a half hour and reset the system.
An entire scared floor out of commission. What else can go wrong?
Hey, where's that?
Okay, so that was a video from Monsters Inc. But I usually use that as an example to show, like at our laboratories, are we really prepared if something happens? Someone who's handling it, suddenly there's a spillage or whatever, are we prepared? So if you are handling GM material, you have to be, okay? You have your equipments in place. You know what to do, who is supposed to do what, how do you do the reporting? Everything must be in place before you start your activity. Okay, so that's why part of the form of what you are filling up in your Form E is related to emergency response plan. So just now we spoke about an incident in a laboratory. What about unintended release? Again, I recall our biological diversity. We don't want anything to go out into the environment and cause, any irreparable damage. So this incident actually happened in 2016. This is Federal Highway. This is one of the main roads in KL. Okay, so it connects KL to PJ. So it's really a very busy road. In June of 2016, there was an ostrich, okay, burung unta. It was running on the highway, terlepas, okay. It somehow escaped from a car and it was running in the highway. This is not a GM ostrich. This is just a normal ostrich, nothing. If it can happen to a normal ostrich, okay, a normal ostrich that made a getaway, ran away, how much more should we be concerned when we are handling GM material in our lab? We need to make sure that everything is in place. Even if something happens, unintended release as we call it, terlepas, what are you going to do? How do you get it back and have the minimal damage and management of the situation? So ask yourself, are you prepared for any unintended release? The third component, okay, we are dealing with a lot of chemicals and all that. This again is not GM material. It is just an incident. Again, it happened in 2016. I got it from the newspaper. Hazmat rushed to Merchant Clinic. It was a mercury spill, okay? So Hazmat came and cleaned up and all that. So, but just say, if there is a spillage that happens in your laboratory, do you know what to do? Okay, do you have a spill kit in your lab? Do you know where it's kept? Has the materials in the spill kit been checked? Is everything in place? Do you know what is the process of cleaning up a spill? So these are all the things that you must take into consideration before you actually handle a GM material in your lab. Okay, I will move on. You need to know what information to submit and how to do it. I think maybe we only got Dr. Cha's notification from UMT. My memory might fail me, but I think I remember only one notification from UMT. There might be more, I am not sure. You can correct me if I'm...
Only from Dr. Cha project so
Okay.
far. All right.
So he has had the experience of filling up Form E. So just to give you an idea, because just now I mentioned, right, two regulatory processes under the Biosafety Act. One is approval and one is notification, okay? So we will go for release activity, okay? Release activity and import. That means there's some sort of contact to the environment. When we look at field trial, you see the first brown box. Depends. Is your GM material a plant or not a plant? If your GM material for field trial is a plant, you fill up Form A. If your GM material for field trial is not a plant, you fill up Form B. That's it. For field trial, these are the two things you have to remember. if you are doing a release to the environment, which is not a field trial, for example, just now we spoke about products or food that is placed in the market. So any of those applicants, theirs is considered other than field trial. So then we will see, is the product from a plant or not a plant? If it's a product from a plant, then you fill up your form C. If it's not a plant, then you fill up form D. So that's it. A, B, C, D. So and all this is a regulatory process for release activities. Okay, field trial or not field trial. Plant or not a plant. That's how we divide the forms A to D. Now we move on to the next kind of activity, things that you do in your laboratory. Contain use activity. If you saw my bar chart earlier on the decisions of the board, a bulk of it is form E. A lot of processing that we did was for contain use activity. Many researchers in Malaysia are working still in the laboratory phase. So you can see most of the things that we are working on is in form E. Export of LMO, also we have some collaboration, sometimes material and things like that. So that would be form F. So these are the things you have to remember. If you're not sure which form to fill up, and it will give you a clear understanding. If you go to our website, we also describe which form is for what. Okay, so you have some assistance there as well. Okay, now I want to zoom in to contain. If you are going to do an experiment, you will start in your laboratory. So for your notification, you want to inform us that you want to work in your lab, you want to do modern biotech. So these are the parts of the form E that you will be able to see. First, we have provide a checklist for you so that you can check to see whether everything are there. Preliminary information, basically this one would be like what is your name, your phone number, how to contact you, those kind of things. Then we also ask you information about your personnel. Personnel means, as you know, nobody in research works alone. Usually you have a team, okay? Maybe you have your master student ke, your PhD student ke, your lab assistant ke, siapa-siapa lah. The basics of why we want to know is we want to know who is going to be handling the GM materials. So we'll ask you for the name and probably like, you know, what are the qualifications of the person in your team. So we will ask you for details about the personnel of your team members lah. Secondly, project description. What is it that you want to do? So this one you can ciplak from your project proposal lah. Okay, this one I want to do this macam ni. My main objective, sub-objective, sub punya objective semua ada lah. Okay, so your project description, that's what we want to know. Next, you go for your details of the LMO. We already saw what you want to do. But specifically, we want to know what is the LMO that you are dealing with. What is the parent organism? What is the sequence that you put in? Where is the sequence from? So these are the kind of information that you need to provide in the table, which is usually table 2 in your form E. Details about the LMO that you are handling. After that, you go for your risk assessment management. Earlier, I told you, right? You have to fill up the table. Okay? Maybe you might have to go through some of our guidelines to fill up how to fill up. But we do provide for your information. If you go to our website, kita ada borang contoh. So you look at our borang contoh and then you can see an example of how to fill up the table. You can also look through our guidelines, or if you're not sure, you can consult your IBC or even consult us for some guidance on how to fill up. Because I cannot do it now and today because it is actually training on its own. Okay. So what else do we ask? We also ask you information about the premises. For example, tadi we use back our same example. If you are going to produce it in your lab A, where you are doing your transformation work and all that, your petri dishes and all that, you're going to keep there. And then after that, you're going to put in your University Malaysia Terengganu transgenic glass house. Okay, you want to transfer kat sana. So what you have to provide in your notification form is details. Okay, your lab ni kat mana? Okay, aras 4 of your bangunan something, Faculty of Science something, those kind of things lah. And then we also ask who is the lab manager, contact details and things like that. And your transgenic glass house, maybe lab manager tak sama dengan manager of the transgenic glass house, right? So dah lain dah. So you fill up the table. Basically, this is the information that you want to know, that you need to provide for us because we want to know, okay? Just one more thing about premises. You don't have to use your own premises. Just say you have collaboration with another university nearby. They have the facility and you don't. You can also use their premises. Apa syaratnya? Syarat is that their IBC must agree for you to bring your GM material over there. If their IBC kata tak nak, you don't come and do here, okay, then too bad for you lah. We also cannot do anything if they don't allow you to bring your GM material there and use their premises. But mostly from our experience, there's already collaboration in place between various universities. They use each other's premises because sometimes they might not have the equipment or the facility ke apa-apa, those kind of things. It's not limited to only premises that is owned by University Malaysia Terengganu. You can use other people's premises, but our qualifier is that we need a letter from the IBC from the other institute to say that diberi kebenaran untuk menggunakan facility. Okay, so that's one thing I wanted to highlight. Your IBC assessment tadi I dah cakap dah. IBC has to do assessment and we want to see the IBC assessment before you submit your information to us. Another thing is the additional information. I gave you some examples just now. You say you're going to autoclave, tapi SOP autoclave tak ada. So those are the additional informations that I mean. If when you submit to us already lengkap selengkap-lengkapnya, there's nothing else for you to add, then okay lah, tak ada lah additional information. But if we do find something that can memantapkan your documentation, then we will ask for additional information. So the next thing I want to talk about is CBI, confidential business information, but I will talk more about it in the next slide. Okay, let me show you the flow. The flow. First, you have to fill up your Form E. We will request 1 original Form E and 6 copies. Currently, this process is a bit stagnant because we are currently working from home and we are facing some challenges on how to receive physical forms. And I believe some of the labs, from what we know from the IBCs who inform us, researchers that we are dealing with also told us that some of them, work at the lab has stopped for a bit because of our current situation. So I am going to talk about processes as if nothing has happened and this is the procedure in place. Okay, 1 original form, 6 copies, submit to the Department of Biosafety. And then any additional information. For example, you kata, okay, I'm dealing with this microbe. Actually, this microbe is very low risk. See, I have 10 publications for you to prove to you that this is a very low risk organism. Okay, just submit it as additional information. Okay, currently we do request for a CD. I know this is like very zaman purba punya requirement because I think the newer laptop CD drive pun tak ada. So, but currently, some have provided us in the form of a thumb drive, but we do require a soft copy version. Why we require a soft copy version, I will explain to you shortly when we talk about the confidential business information. Submission of Form E, no need any fees. So you don't have to hantar duit kat kita lah, memang is zero fees, okay? IBC assessment form, it is a requirement, okay? So what happens? You, the researcher, the applicant, dah hantar one part. Bila IBC look at your form, say, okay, we need to add some more information, they put in together the additional information. JBK say, document ni tak ada, okay, then we fill up. Then GMAT kata, okay, fill up. So everybody is working together to get a piece of the puzzle. The puzzle is your activity, the description of your activity that is going to go to the board for the assessment. So we want to make it to the best of our ability, as much information and all the issues that surrounds the activity are already addressed. So that masa sampai kat board, senang je. Mereka tengok semua assessment dah lengkap, lulus je. Itu je. Okay? So it is an interactive process. It doesn't happen like, okay, dah sampai satu orang dah, after that kita duduk diam je, you never hear from us. So it's an interactive process that happens. Okay? Let's talk about confidential business information. We do have a provision under the Biosafety Act. whereby when you give us information, you can claim it as CBI. What is CBI? Information that is not generally known among the circle of people who might have access to this kind of information. Maybe you have something for us, a procedure that you did in your previous experiment that you're going to do in this experiment as well. But maybe it hasn't been published yet. No problem. So you just declare it as CBI. Okay. Or the information has commercial value, maybe some process that you have already patented and then you want to keep into a secret, or you have taken some reasonable steps to keep the information secret. You don't declare something that is CBI, tapi tomorrow you put in your blog site, guess what? I already have this, blah, blah, blah. Okay, so that is not CBI, okay? Something that you keep in secret until it is ready to be made public access. Okay, that means you yourself have done reasonable steps to keep the information secret. Okay, so what do? You mark it as CBI and then we will treat it as CBI. Remember, your notification form is going to go to GMAC. All GMAC members have signed up a form to say that whatever that we are discussing in our GMAC meeting, all documentations that we send to them, everything is confidential. Okay. So this following information is not considered confidential, name, address, and all that. So I think this all would be relevant if you are going to do a field trial and then you're going to produce a fact sheet, remember? So all this will be in your fact sheet. But as I mentioned earlier, if you're doing a notification in your notification form is not publicly accessible. It's only accessible to the pegawai-pegawai in the Department of Biosafety. It is accessible to our GMAC members, accessible to our board members, and maybe some regulatory agencies who might be involved. Other than that, information does not go out. So you can rest assured that all information in your form is kept confidential regardless. Right. So let's talk about your IBC. Yeah? It's very important for you to understand apakah fungsi IBC ini. Right. IBC. So now I've moved to the introduction slide of the IBC. IBC is a formal expert committee. This group of IBC in your University Malaysia Terengganu, they didn't suddenly come together, jom kita buat IBC. Nope, it's not like that. Okay. The management of University Malaysia Terengganu appointed them to be the IBC. What is the function of the IBC? Function of the IBC is to monitor modern biotech activity at your institutional level. Okay, and to ensure compliance to the Biosafety Act 2007. Okay, siapakah IBC? You have your IBC chair. Tadi dengar kan Prof Nur Nawati, right? And then you have Azlina as your BSO, and then you have your IBC members, right? So these are the main people in your IBC. Get to know your IBC. It's really important. I have very little time here. I will just highlight certain functions of the IBC that might be of interest. But if you are interested to know actually the full function of the IBC, you can read the guidelines for IBC that we have on our website. Another thing about the IBC, under the Biosafety Act, the function of the IBC is to monitor Biosafety Act 2007 and ensure compliance of all the requirements under the Biosafety Act. But management of University Malaysia Terengganu has the authority to extend the scope of IBC. It's up to them. The basic function requirement is compliance to Biosafety Act. But if University Malaysia Terengganu said, okay, you guys IBC, you also have to handle biosecurity issues. You guys IBC, you also have to handle occupational and safety health. Okay, that is your internal arrangement. Okay, don't contest the IBC and say, no, you see Regulation 5 said Biosafety Act only. Why you sibuk-sibuk about other things? No, the basic function is compliance to Biosafety Act. But if University Malaysia Terengganu decides to expand the scope, that is completely management decision and you have to comply regardless. Okay, right. So IBC responsibilities, as you can see on my screen, there are 4 basic functions of the IBC. First is to provide guidance for safe use of modern biotech, monitoring the activity. Okay, don't be like, why they simply want to see what I'm doing, like inspect my lab, tengok my SOP. Why? Itulah fungsi dia. See, number 2. That is their function. They are doing their job. Okay? That is the function of the IBC. Number 3 is to establish, monitor implementation policies, procedures. This also includes like what we are having currently right now, awareness activities about Biosafety Act 2007, compliance requirements, capacity building. All this is IBC punya kerja. Okay, they've already organized one today. So I hope you have the full benefit of this event. And finally is to determine the biosafety level for the facility. So whatever facility that you are using, so IBC will take a look at it because we actually value inspection reports by IBC for all the premises that you're going to be using. We will look through the IBC inspection report. IBC might say that, hey, this facility, I think it cannot be BSL2, like it has to be BSL1 because it doesn't have this. So IBC will say, in dalam hati, you say, no, in my heart, I feel this is BSL2. Okay, fine. In your heart, that's it. But if your IBC decides that your facility is BSL1, then it is BSL1 and we will go according to what the IBC has decided. Okay, they are mandated to do that. They have been given authority. You tak percaya, go to regulations 5. It's written there for you. Okay, you saw this slide earlier just now, overview of a 3-tier risk assessment process. I want you to see exactly IBC fits in there. Kalau tak ada IBC, borang tu takkan datang ke JBK. Okay, and also they play a fundamental role in making sure that all your documentations are complete and good before it reaches the Department of Biosafety. Okay. So you have your risk assessment, management, emergency plans, and they will also look at that. What does an IBC assessment mean? Okay, IBC assessment means that they give you an endorsement. So actually, when they are looking through your borang E, looking through your SOP, looking through your training records of your team member and all that, they are actually IBC filling up Annex 2. Annex 2 will be submitted to the Department of Biosafety along with all the other things that you have submitted to the IBC. So kalau dia dah lulus, they don't find any problems, then we also will take into consideration, okay, IBC has already looked at it. If we find that IBC assessment report is missing, we will tendang balik to you. Please go through your IBC. Okay, kalau tak ada IBC, the borang is not going to be processed by the Department of Biosafety. Right. So I'm going back to this slide whereby your risk assessment must make sure that the premises being used is suitable. So IBC's job will be to ensure that the facility is suitable for the activity. Okay. Personnel assessment. So we are sitting here in Putrajaya. You know, we don't know who are your team members. Of course, we can provide record of training and all that if it requires. Tapi kita tak kenal sangat who are the personnel who are involved in your team. Contohnya lah, just say you are Avengers punya team lah kan, right? So these are your team members lah, all of them, okay? So we are fully dependent on IBC to do the assessment of the personnel involved, whether dia sesuai ke tak, okay? And we are especially concerned because now you are also a university and many students might be involved to do their master's project, their PhD project. They will be involved in your research activities. So we are specifically concerned for students to make sure that they are properly trained before they handle any activity. For example, they must be well aware if you are handling GM material, there must be a specific SOP for disposal. Kalau tak, dia buang je or throw it under the, buang in the sink or something like that. So we are specifically concerned for students, but you as the IBC will check and see, okay, is this person suitable? Okay, is this person suitable to work in a lab? For example, macam Avenger ni, Hulk ni asyik marah je, okay? And then he's also big size. Do you think he's suitable to work in your lab? I don't know. Let's see what the IBC says, okay? So these kind of things are especially critical for high-risk activities. For example, if you are working with a highly pathogenic organisms, maybe like lentivirus or something like that, or if you are involving animals. Some examples of animals, because sometimes you will handle sharps in terms of like, you will you will handle a syringe. So disposal of sharps is different from other liquid materials or something like that. And we want to know that the person who is handling it is properly safe. So personnel assessment, we are heavily dependent on IBC. On top of that, sometimes you will also require macam record of training or something like that for any specific personnel who is in your team members. Okay. What else does the IBC do? Inspection of premises. I said just now, right? They also do inspection report and they submit to us. So just say kita guna balik contoh tadi tu kan. So you're working in your lab, you are developing your GM material, and then from there, then you go to your University Malaysia Terengganu Transgenic Glass House. Okay, so you have two premises, right? So IBC will give us two inspection report. One is for your lab, one is for your University Malaysia Terengganu Transgenic Glass House. Two inspection report, okay? And then we want the inspection to be as recent as possible, within a year that the form has been submitted. You cannot submit, okay, your inspection report, tapi dia punya inspection done in 1985, masa tu, sorry lah, it's okay, no need. Please do a recent inspection report and submit it to us. Currently, with the pandemic situation, we are a bit flexible because we know there's a delay in a lot of research work that is being done. So we are trying to be a bit flexible in terms of, within a year, because a lot of things haven't been moving. So perhaps, if you do have within 1 or 2 years, it is still acceptable. Okay. So IBC needs to inspect the premises used at least once a year. Sometimes if your project is a 3-year punya project, so your first year, IBC already did the inspection before you submitted the Borang E. Second year and third year, IBC will come and do the inspection. Don't be like, hey, that's why already done what last time. Why sibuk-sibuk nak buat banyak kali? No, it is a requirement for them to inspect your ongoing activity at least once a year. Okay, it's on a case-by-case basis, so depends on the risk level of the activity. Okay, maybe they feel that once a year is not enough. Maybe we want to inspect twice a year, or sometimes maybe there's an incident in the lab, so they want to make sure that there's no compliance issue or anything like that lah. They can decide to come more than once a year. Okay, don't be upset lah. No, if they feel they need to come more than once a year, they can. Okay, boleh je, tak apa. Right. So the next thing I want to talk about is, you should see on my screen, standard operating procedures. Okay, this is something really important and we emphasize a lot on this. The SOP that you produce for your risk management strategies. Contohnya lah, we go by the example that we are planting something in your University Malaysia Terengganu Transgenic Glass House. Okay, you are planting there, but then you ada SOP lah that you want to autoclave. So you have to come up with an SOP, and then the SOP must be approved by the IBC. You cannot suka-suka do your SOP and then submit directly to us, cannot. Your IBC must agree that yes, this SOP is good and it works and it can manage the risk of unintended release from your transgenic glass house to the environment. Boleh, okay, so they will endorse and then they will give it to us. SOP also, similar to the inspection report of premises, cannot be more than 2 years. You must revise your SOP periodically. Okay, so you cannot pakai lah SOP, maybe now you SOP from 1960 pula tu, SOP, then you submit to us, cannot. SOP must be revised frequently. The act of revision might not necessarily result in any amendment of the SOP, yeah? But you still have to look through it to make sure it is still applicable. Okay, but one thing that you must take note of, okay, there's a typo on my slide. Once you submit the SOP to us, you cannot make any changes, because remember the flow chart, right? Once you get your letter of acknowledgement, you can start your activity. When you start your activity, what you are doing is following all the SOPs that you have already submitted to us. So when we are doing the assessment at the GMAC stage, at the board stage, we know that this is what you are doing. After you submit to us, you cannot suddenly change your mind and say, no, I don't want to autoclave, I want to do chemical punya treatment for my disposal, semua tak boleh. Okay, you just stick to what you have written and do it accordingly. Okay, a copy of the final version, because sometimes when we look at the SOP, we might find some changes because the SOP is not event specific. Just say you are working in, I use back the same example again and again, okay? You are planting your GM material in your transgenic glass house. We look at your SOP and then suddenly we say disposal of sharps. The syringe will be put in a, the syringe that we use will be put in a sharps container. Apa pula ni? You are working with a plant and then you talk about a syringe, okay? So your SOP must be activity specific. Then you will find lah surat from JBK. Please revise your SOP to be activity specific. Keluarkan cucuk-cucuk ni from your SOP. Only stick to the material that you are using, which is a plant material, that you are not using a syringe. Okay, so you'll get a letter from us. You revise the SOP and then the final version is the one that must be kept by the IBC and also at the site and the premises. Don't put your original SOP kan, that you didn't revise yet. The final version that is going to be used once the revision has been done either by request by JBK or GMAC, that is the final version of the SOP that must be used. Okay, communication with JBK and the IBC. Okay, if you are not sure about anything, any clarifications, discussion, if we ask for any additional documents, macam tadi saya bagi a few examples, right? We want SOP ke, we want inspection report and all that. We will only liaise with the biosafety officer. Usually, we will always talk with the biosafety officer. Once the biosafety officer, sometimes the biosafety officer will say, I think you better discuss with the PI because the PI can explain better, okay? Because your biosafety officer might not have the expertise or fully understand what is the activity that you're doing, okay? Then we will discuss with the principal investigator, the researcher, which is you, the applicant, okay? We will not discuss with team members. Okay, you cannot say, okay, discuss with my lab assistant, or discuss with my PhD student for flat, very good. Okay, totally fine. But in terms of communication, we only liaise with biosafety officer or the principal investigator at the list. Other than that, other team members, we do not interact with them. If they say anything to us, we are also not concerned because the final say will be the principal investigator or the biosafety officer. Why? It's not that because kita sombong, but it comes for accountability of information. Okay, one team member can say like that, somebody else can say something else, and then we don't want to deal with irregularity of information, because you must remember sometimes you might be dealing with SOPs, or for example, your SOP has how many percentage of the chemical that you are using, all these kind of things. We want you to be really sure, so that's why the line of communication is very specific. Okay, so IBC monitoring after the NBB decision. Okay, so IBC will keep a record of the relevant NBB form, your Annex 2, which is the assessment form, risk matrix, and whatever that we have given. Okay, first review of the approval will take within the 12 months. If so happen lah, your activities are short term, maybe 9 months dah habis, mungkin tak perlu lah. But the first review will take place within, sometimes it's a 3-year project, 5-year project. They will review back your activity in the 12 months. Okay. Subsequent review will be done after a period of 1 year. So all IBC monitoring activities are documented. For your information, IBC has an annual report that they submit to the Department of Biosafety once a year. Okay. In that report, they will say, okay, these are the activities that are ongoing. These are the activities that habis already. These are the inspections that we have done for all these facilities. These are the materials that are being stored currently at our facility. These are the capacity building activities that we have done. So all these kind of things, they will report to the Department of Biosafety once a year. So let's talk about extension, termination of activity period. Okay. So once you have finished your project, your research work is done. So we are talking about research. Sometimes the material that you produce, maybe you want to work at it at another angle. So maybe you want to continue working at it, but you don't want to produce the GM material, a new one. From here, you want to do the second part of the experiment. Fine, you can store it. But let's talk about a situation where you have not finished your activity. Just say I said I want to finish by December 2021. This is very relevant right now because a lot of researchers are struggling to meet their deadline. Okay, So what they need to do is they need to submit a project extension form. For your information, if you want to extend, it's totally fine with us. As we know, with research, a lot of things can go wrong. Even if without the pandemic, sometimes your autoclave not working. Maybe your chemical tak sampai, and then suddenly your master student quit. Macam ni lah, macam-macam, all types of scenario. And then on top of that, now we have the pandemic, MCO, PKP, cannot go to the lab, this, that, everything. Okay, so sometimes you might want to extend your time period. For JBK, is totally fine with us. For JBK, the time period, what we want to take note is how long you are doing it, because we are monitoring your activity. So if you dah habis, you want to know dah habis. Tak adalah you dah habis activity, then kita pergi Universiti Malaysia Terengganu nak tengok your notification, your activity, what is being done. Nak tengok, dah habis lah. Okay, we don't want something like that. Okay, so for us, is monitoring is for oversight. Okay, but for IBC is also oversight as well, because they are also doing a more thorough and regular inspection and monitoring of the activity. If you feel you cannot finish on time, then you just fill up your borang Annex 5A. You tell IBC, I cannot finish on time because of this. I need to extend my timeline. Boleh je. Okay, so but you have to inform your IBC. Okay, when IBC gets Annex 5A, they will submit a copy to us. So you no need to give it to us. IBC will inform us that this activity has been extended. Okay, don't besok nak habis, then baru today you want to fill up your Annex 5. Janganlah macam tu. Okay, have some sympathy for your IBC. Okay, your Annex 5A, at least fill up 1 month earlier. More or less, you already know what you are going to delay. Takkanlah hari ni baru tahu you're going to delay, right? It's not like that. So you fill up earlier. Have some sympathy for your IBC, because you usually in a university setting, the function of IBC members is not only to be an IBC member. Okay, usually they are lecturers, they are also doing their own research in the laboratory, and then dah lah jadi IBC member pula tu. So have some sympathy for them. Okay, cooperate with them. Okay, show some compliance of whatever that is required of them. Okay, cut some slack for them, because they are also working hard to try to comply. At the end of the day, what is the thing that you get? Universiti Malaysia Terengganu is totally compliant to the requirement of the Biosafety Act, right? Everybody wins, right? Okay, so a copy of the approved form should be submitted to the IBC 1BC. Don't give them last minute. Please don't give them additional headache. Now, termination form. Just now I spoke about the material that you want to use for your subsequent activity. That is totally fine with us. You just fill up your notification, your termination form 5, Annex 5B. Okay? Why is a termination form so important? When you finish your activity, you are no longer using the GM material, you are required to dispose of it. That means you do the treatment, autoclave, whatever, and then you dispose of it, it doesn't exist anymore. If you want to do it for subsequent experiment, you want to store, that is also fine. Fill up the relevant form 5B. Why is it important? Especially so in a university setting. How many of y'all have your own lab? You sorang-sorang duduk that lab. Minus 80, all yours. Minus 20 freezer, all yours. You tak payah share dengan siapa-siapa. Everything you punya. It doesn't work that way in normal laboratories. Usually you share your minus 80 freezer, you share workspace and all that. So sometimes, especially in a university setting, the turnover rate is high. Your master student has graduated, PhD student graduated. Suddenly, when you open your minus 20 freezer, buat gotong royong, siapa punya ni? Nobody knows, okay? Buang, okay? So we are concerned of these kind of situations. We don't want GM materials to end up like that. So there must be some sort of accountability, okay? So if you have storing something, you fill up your Annex 5B. If you are no longer storing it, you fill up your Annex 5A and submit to the IBC. You want to store, not a problem. Just that Annex 5B, you just have to bagi detail lah, store kat mana, siapa in charge, berapa banyak you store, those kind of things. Okay? Next, I'll go to resources available about biosafety. This is how our website looks like, www.biosafety.gov.my. All our material is available for download, free of charge. All is available in PDF format. Just now I said that today my presentation is basically on research and what would be of interest for researchers. But the Biosafety Act does cover more than that. And if you are interested, I suggest you read the book on the top left corner, User's Guide to the Biosafety Act and Regulations. If I don't know whether you have seen any, Biosafety Act or, any other legislation, those are very lawyer punya language, panjang-panjang dia punya ayat. You start from the beginning, by the time you reach the end of the sentence, you don't know what the front has said. Okay, I'm not a lawyer by training, so I also sometimes find it a bit difficult to understand. So this book actually puts it in layman language, exactly what are the requirements of the Biosafety Act and its regulations. You can take a look at that. Another thing I would recommend for you to read is contained use activity. So basically, it gives you some specifications of laboratories and practices of the laboratory, how to dispose, and those kind of things, okay? And then if you're interested to know about the IBC, so this is the guidelines on IBC. The functions of the IBC and all that is available on this website. Okay, so the last thing I want to talk to you is about there are some activities and techniques that are exempted under the Biosafety Act. Exemption of the Biosafety Act is provided by the minister. List of exemptions may be revised from time to time. Okay, depends. Okay, so we know a lot of things about this. Then after that, if we know it's already low risk, you have the history of safe use, then you can add it into the exemptions list. Okay, so I give you some examples of exemption. So export of LMO, because most of us are research here. So we go back to our example. Okay, our corn to Argentina tu kan. Argentina, you want to export your GM corn to Argentina, to your collaborator, University of Argentina. So just say I already sent, I fill up my form F, JBK dah approve, I already sent my first batch. Mana tahu, at University of Argentina, blackout ke apa ke, all your materials finish, no longer available. So then your collaborator ke Argentina kata, hey, can you send me another batch because something happened lah, my lab all material lost already. Tak apa, it's okay. So exemption is same as LMO, same country, same purpose. Okay, we already get a letter from us to submit, to send your LMO to Argentina, to the same university, your same LMO, go ahead and send. You no need to fill up form F again. Okay, so this is an example of a simplification process for exemption. Okay, the next exemption I want to talk about is subsequent approval not needed for LMO that is for food, feed and processing. So if a developer of this GM corn already approved to be placed in the market, you no need to apply for another approval. That means anybody else can bring it in. Third example of exemption is like just see if my baju here is made out of GM cotton, I no need to fill up my borang C to get approval to wear my baju. No need. Or if my table here that my laptop is on is made out of GM wood, I do not need to fill up borang C and get approval for this table. So GM wood, GM cotton used for clothes and furniture, no need. Why? Because it's of low risk. You cannot lah have one GM kerusi ni jadi 10 GM kerusi, right? It's not going to impact the environment and all that. So that is why we consider it a low risk. Okay, let's go to the exemption of first schedule. So this one will be quite relevant to most of our researchers here. For you to know that there is a first schedule under the regulation whereby we provide exemption for contained use. So it's not for open release, it's for contained use. Certain techniques, certain organisms that you are using, as long as it meets the conditions for the exemption, it is exempted. So examples are these are very well understood organisms and possess very low risk. Okay, we know a lot of information about that, so we no need to regulate under the Biosafety Act. But if your activity is exempted, what will happen? If you want to know more about the exemption, again, this is our website. You can go here and you can see the exemption list. Okay, this is our website. You can see the full list of exemptions here. Okay, so if you find out that you are doing modern biotechnology, okay, if it's a field trial or whatever, you go through your IBC and then you fill up your relevant form. If you're going to do contained use and then you tengok your first schedule, check for exemption. If it's not exempted, then you go by the same process as I mentioned earlier. Fill up your form E, go to your IBC, and then it will come to JBK, GMAC, and then the board. However, if you find out that your activity is exempted, it is not regulated under the Biosafety Act, meaning that you tak perlu isi borang E. but you still have to inform your IBC. Remember earlier I mentioned that IBC will always be sending us annual report. One of the things that they are reporting is they are also giving us the list of exempted activities that are taking place at your facility. Okay, so they are doing the monitoring and reporting, meaning that JBK is not going to be involved. We are not going to come and look at your exempted activity. We leave it to IBC to do the monitoring and they will report to the board. So that is what's happening to exempted activity. Okay, so that was a long 2 hours that you heard from me. Summary of what you heard. So hopefully by now you can at least know what the Biosafety Act is all about. By now you should know what NBB stands for, National Biosafety Board. You heard about the two regulatory processes under the Biosafety Act. You know how to get an approval from the board when you want to conduct a research, what kind of research, what kind of form to send in. You heard very briefly how to do a risk assessment and management and emergency response plan, why it is required. You know what kind of information, at least for Form E, what is required and how to fill it up. And you also learned, for me, this is the main component, IBC. How does IBC fit in the picture of all this? And then after that, I just pointed you out to some biosafety resources that we have on our website, and also very briefly about exempted documents. Okay, so 2 hours we have spent together, you and I. If you're still not sure about what you heard, you're confused, you don't know what to do, please don't worry. You still have your IBC, you can consult, or you can drop us an email at dob@biosafety.gov.my, and we are happy to assist. Okay. So I think I will pass back the time. So we have all the social media platforms, as you can see. Take note of them. Why is it important? Because as I said, perhaps you can join in the next public consultation that we are doing. You can join us, you can like us on Facebook, you can follow us on Twitter and Instagram. When you know all this, when you're following us on all the social media platform, then you can also follow if there's any public consultation being done. That's an opportunity for you to participate. All right. So thank you very much for your kind attention.
Thank you, Dr. Anita. I think it's really very comprehensive presentation, which cover many topics. And especially for the second session, I think you highlighted the Form E and how to fill out the Form E, all that, and all the procedure. So it's really very, very important for researchers that are involved in modern genetics. So for researchers that you are not sure, if you have doubt whether your research works that have been characterized under this modern genetics, and that you need to submit your notification. If you are not sure, I think you can just contact our BSO, Biosafety Officer, which Azrina. All of you, I think you know her already. So for your information, IBC in UMT, we are called IBBC. So with expanded responsibility in IMB. So IBBC in UMT is called IBBC. Okay, so we open for Q&A. If you have any questions to ask Dr. Anita, please. Any questions? If you don't have, maybe I would like to ask one. How about the enforcement for non-compliance now, Dr. Anita? Is there any cases that researchers have been fined for non-compliance?
Okay, so if you recall, there was one regulation for compounding, if you recall. So we already have a regulation for compounding. We have a legal mechanism to compound researchers who are non-compliant. On top of that, we have also regularly sent warning letters to the institute to inform that this researcher has not complied. So these are some of the mechanisms that are being done. So far, we have not used the compounding regulations to take action or penalize any researchers. We actually have a soft heart also in terms of like, we would first approach is try to encourage and compliance. But then again, it's already been nearly 10 years that the Biosafety Act is already enforced. And by now, we would expect researchers to be aware, or at least institutes to be aware of the requirements under this. So I think it won't be long if, as the years go by, how we implement is becoming stricter and stricter. Like, for example, like 10 years ago, we saw, oh, you're doing activity. Okay, actually, you have to fill up Form E. Okay, baik-baik, you bagi tahu macam tu. But then after now, straight away, you get warning letter to your head of institute to say that this researcher, has been doing activity but has not complied to the Biosafety Act. So, you can see the stringency of the enforcement is becoming tighter and tighter. Okay, so back to Dr. Chow, we have not taken anyone to court yet, and the compounding regulations has not been used on any researchers yet, but I would strongly advise not to get too comfortable.
Thank you. I think we don't have question in the chat room. Okay, so if you don't have any questions, maybe Azina, we end the session, is it?
We have Prof. Azna with us.
Oh, okay.
So the IBC chair, Prof, you want to say something to Dr. Anita? We met her, I think, few years back.
Yes, yes.
Silakan,
Hi,
Prof.
Dr. Anita.
Hi, Prof. Good to see you again.
For so long, I guess it's like more than 5 years. Yeah.
Definitely more than
And
that.
I would like to say thank you to you for a very detailed presentation. We call presentation. And I guess everyone who involved in this, they will understand the most, especially Dr. Cha. So the Biosafety Act, we keep talking about this, I guess, for a year already, Azina. So we hope that everyone who work with this, then they understand, and they. But our problem is now, our researchers is actually quite shy to declare. So we cannot just ambush and make something out of it. So we hope that everyone at UMT will declare their research all about. Up to now, we propose to our CREAM Research Center so that we can actually evaluate from the beginning of their project before the approval of any of the research grant. But up to now, it's not happened yet. But we keep telling our research management center that we have to do something about it. But thank you very much for very, I guess, a very good information to our all researchers at UMT. And we hope that next time we can have another banker. That's my suggestion lah. Because, when you talk like more than 2 hours today, people keep on nodding their head, showing that they understand the most of whatever of your presentation. But actually, when you didn't do and hands-on of this, then perhaps they might forget about this after the slot. Yeah. Thank you very much, Dr. Anita. Thank you very much, Azina. And thank you very much, the moderator, for this session today.
Thank you, Prof.
Okay.
I think before I end the sessions, I would like to highlight one more point. Is to all the researchers, to remind all the researchers, because we have to aware that maybe let's say your research is exempted from in the list for exemptions, but you still need to notify IBBC, UMT.
Yes, yeah.
Need to notify. You need to declare. This is very important. Okay. Thank you. I think before I end the sessions, I would like to thanks again to PPBI and CREAM for the support and organized this webinar. Okay, thank you, Dr. Anita, and thank you all to all of the participants. And you already spent, I think, more than two and a half hours. Okay, thank you very much. Thank you. We should end the session now.
Thank you very much on behalf of the Department of Biosafety for inviting us, Dr. Cha, Azlina, Prof. It's a great opportunity for us because, like, usually we would physically go to your place and conduct training and awareness and all that, but it was very well organized. Really salute your organizers and thank you really for providing the opportunity for this. Hopefully, we are just for the IBC member, a word to the IBC members. We are here for you. Anything you want to consult us, we are always here. Okay, just consult us and then maybe we can work together on how to maybe improve things or anything like that.
Thank you, Dr. Anita. I think the most important part to help with the researcher is how to carry on their risk assessment. Based on my experience conducting Dr. Cha's risk assessment project on behalf of IBBC is quite a headache task. Lots of documents and journals to refer to. And I think further workshops we will discuss on the risk assessment. And my concern is about the field trial or the greenhouse or the aquaculture parts lah, because we are dealing with the aquaculture part research part as well. So hopefully in the next session we will discuss on that further. We'll contact you later. Okay. Thank you a lot. Thanks a lot, Doctor, for the help from the JBK. I remember Raimi was helping helped me with Dr. Cha's application as well as Azarina now as is on Azarina. So thank you a lot. Thanks a lot. See you around. Okay, bye. Bye, Dr.
Everyone. Thank you for listening.
Take care. Terima kasih juga
Okay,
semua.
bye.
Bye.